तो वो तो पीपीटी में चाहिए कितना हो गया? कौन? कोई? पहले लो। अंग्रेज आप लो। दे दो। कर दिया मैंने जी। सब मेरी नहीं। नहीं देखी। ओह। तब मैं मेरे को कॉल कर दी। ग्रुप है। कौन कौन है? रेडी। कॉल। सुबह सुबह सुबह। पर्सन है। कुछ फैमिली के लोग आ रहे हैं ना, कोट पहन के लंबे लंबे लोग। फिर फील होता है कि कितने छोटे हैं ये। सब सीरियसली। बाहर नहीं है, बाहर नहीं आ रहा। ज्यादा मैंने तो मैच रहा है। पर अभी मैं खाई नहीं खुद ही। फोटो तो मुझे खींचना है। मैं खोलना पड़ेगा। आगे का मुझे नहीं पता। चलो, खाली से बैठो, एप्पल भी ले रखे हैं तो। अरे मैं फोन से खींच लूंगी। तो बोलो ना, लाइक करती थी कि गो टू हेल्प। यूट्यूब पे सर, दीपक का स्टार्टिंग में दीपक ने कहा था, अपना यूट्यूब पे नहीं दिख रहा है। तो यार, शेयर भी क्या बोला इसमें चलो तो एडमिन की लड़कियां नहीं कर देते हैं। वो यहाँ पे यार आईटी की फोटो नहीं ली या फिर जिन्होंने ऐसे 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 भी रहेंगे। प्रेजेंटेशन के लिए आएगी। हाँ हाँ, बोल देना प्रेजेंटेशन है, डाल दी। फोन कर दीजिए। हाँ ममता। हाँ, वो भी तो अंदर अंदर एक। हाँ हाँ। तो
ये सेशन स्टार्ट करेंगे। वो फ्रिक्वेंसी माइक भी तो देखो ना, चेंज है, अलग है। अब वहीं से ऑन करना, उसको पीछे छेड़ोगे ना तो उसकी वैसे ही सेम वैसे नेल से ऊपर की तरफ दबाओ। लॉन्ग प्रेस यार, कोई किसी और को दबाओ। लॉन्ग प्रेस करना है ऐसे।
इसको लॉन्ग प्रेस करना है। बट ये ऑफ भी नहीं कर।
Good morning, all. We'll be starting in 5 minutes. I request everyone to be kindly seated.
ऑफ ही नहीं हो रहा।
और ऊपर की तरफ प्रेस कर दो। ग्रीन लाइट होगी तो खोलना होने।
नहीं ऐसे करो ना, ऐसे पीछे ऊपर की तरफ इसको ऐसे करना है।
स्विच ऑफ कर।
अब बंद बंद भी ऐसे करना है, दबा के रखो उसे।
I request everyone to be kindly seated. We have the chief guest amongst us. I request all the audience members to kindly mute your cellular devices before we commence today's event. A very good morning to all our distinguished guests, speakers, and participants. We're honored to have with us esteemed leaders from various sectors, thought leaders, policymakers, and innovators who are driving change and contributing to India's growth story. In a run-up to BRIC THSTI's 15th Foundation Day celebration, we welcome you all to SYNCHN 2024. Today's session is divided into two parts. The first session will have the presentation and discussion, followed by a vote of thanks. And the second session will have campus tour and parallel networking breakout sessions. Today's event is envisaged to bring together academia and industry to foster stronger ties of collaboration and innovation, which aligns with the government's two most pivotal initiatives, the Atmanirbhar Bharat and Viksit Bharat programs, that aims at transforming India into a self-reliant nation by its centenary year of independence 2047.
THSTI is not just another academic institute. Being at the forefront of translational research, we bridge the gap between scientific discovery and practical applications. We possess the knowledge, infrastructure, and trained personnel required to transform promising research into life-saving products. This meet is a crucial platform where academic insights and industrial expertise come together to foster collaborations, driving innovation, and creating a synergy between research and practical. And then let this be first of many such gatherings where we come together leveraging our strength to create a roadmap that will propel India towards self-reliance and holistic development. Thank you for being here today, and let us begin this significant deliberation with the spirit of innovation and collaboration.
To begin today's proceedings on an auspicious note, we will commence with the lamp lighting ceremony. Please join me in welcoming our distinguished guests on the stage. I request Professor Karthikeyan, Executive Director, BRIC THSTI, to accompany our esteemed guests, Professor Vinod Kumar Paul, Member Niti Ayog, Professor Nirmal Kumar Ganguly, former DG ICMR, Dr. Alka Sharma, Senior Advisor, Department of Biotechnology, on the dais to perform the honors. I thank all the esteemed guests for leading the ceremony, and I request all the dignitaries to kindly take their seats on the stage. Dr. Alka, ma'am, okay, fine, sure, thank you.
Now I request Dr. Amit Awasthi, Senior Professor, THSTI, to kindly deliver the welcome address. Welcome, sir.
Uh, Very good morning, everyone. And it is heartening and inspiring to see such a turnaround. A lot of people are here on the Sunday morning. My special welcome to the dignitaries on the dais, Professor Binod Pal, Member Niti Ayog, Professor N.K. Ganguly, our Director, and Dr. Alka Sharma, Senior Advisor, Department of Biotechnology, and we are expecting Dr. Rajesh Gokhale sometime very soon. So this is a kind of historic moment that THSTI partnering, you know, companies and industries, startups, right, to bring something to the next level. And this SYNCHN, which is having a meaning, SYNCHN means Sinchana, something like, you are reading something like watering a tree, right? So it is having a meaning that synergy in science built through elements of collaboration of high uh, quality data and effective in NCR Biotech Science Cluster. It's called SYNCHN. A lot of thoughts has been put when we come up with the name of SYNCHN. And this platform, SYNCHN platform, is dedicated to fostering in cutting-edge scientific endeavors. Given our institute expertise in translational research and advanced infrastructure, we are uniquely now positioned to catalyze bio-innovation and drive India's scientific progress towards the vision of Made in India, or we call Atmanirbhar Bharat. We aim to strengthen partnership between THSTI and industry, leading to groundbreaking achievement. And I wish all of us here for this exciting Sunday today. And then as you see in the program, we will have a presentation by the scientist on the technology and the platforms which we feel is mature to take to the next level in collaboration with the industrial partner. And then we will also hear industries, their strength, and then we will have breakout session, as you can see in the program, where we can really have one-to-one discussion with each of our industry partner. What is their need? What is our need? And how we can join hand together to now actually help India's biotech sector growth. Because after COVID, this is the right time. Now we are actually pushing this. This is important event. And this is something gonna catalyze for many institution in the DBT and other in CSI and other institution to come up with joint hand together to participate and help India grow Atmanirbhar Bharat. With this, thank you very much. I welcome you all again on the behalf of Executive Director and THSTI family. Enjoy this high energy Sunday. Thank you very much.
Thank you, sir, for your gracious welcome address. Please join me in welcoming Professor Karthikeyan to deliver the opening address to the gathering.
Thank you, Amit, for that welcome address. So it is really an energizing Sunday. I've never seen so many people in this auditorium. So on behalf of THSTI family, it is my privilege to welcome Professor Paul, Dr. Ganguly, and Dr. Alka Sharma here, and all our industry partners, all of you are partners. And I just wanted to say that, see, this is the first academia industry meeting in the biomedical space in this country, at least as far as I know. So I think we are making a very good beginning. And this is, like Amit pointed out, this is the right time to start because we are at an inflection point, both in terms of the THSTI growth story and in terms of the India growth story. So I just wanted to briefly address two questions which are probably in many of your minds. Why do we need to do this at this point in time? And what do we hope to achieve? So why? So I just wanted to show you this slide. So this is just the investment in health research and development in this country, and just comparing it with the US. There is a break you will see in the x-axis for the US. So it's about 80-fold higher. They are a very rich country and we cannot afford to do that much. What we are doing is pretty good, but it is very small. And the reason, if you look at the sources of investment, and the red is public sector, that is government, and the blue is industry, you'll see that most of the research and research investment comes from industry in developed countries. So I think the reason why we are here is this. We hitherto in this country, we have not reached out to industry for taking products which are developed in academia to forward to market. So I think this is the first step that we are doing. And I hope that this will serve as a model for industry academic collaboration in this country, particularly with regard to medical research. We envisage that this will result in collaborations. You will have a whole day to interact, and maybe you will carry forward some of these interactions and you'll continue to interact over the next year or so. And the idea is to bring to market effective, affordable products that will be used in this country. So I just hope that you will all have fruitful interactions and you will develop long-term partnerships, which will be good for the country and good for the institute as well. Thank you very much.
Thank you, sir, for informative and inspirational thoughts. We will now begin with our first session. Under this, the THSTI faculties and scientists will be giving detailed overview on the key strength research areas of THSTI. But before begin, I would like to request all the speakers to kindly stick with the allotted time. To begin with, I invite Dr. Bhaptosh Das, Professor THSTI, to introduce us about the current developments in live biotherapeutics at Institute.
So, good morning to all of you, and very happy to see such huge gathering this Sunday morning. And I'm Bhavatos, working in THSTI almost in last 12 years. So today, what actually I'm going to present what we have done almost in the last 12 years in THSTI in terms of the development of live biotherapeutics. So we know biotherapeutic is a very common term. So if you look the WHO definitions, it's a therapeutics, those have the biological origin. Either it's from the eukaryotic or it's from the prokaryotic. So if you look the clinical industry, the most commonly used biotherapeutics is the vaccines. Then you do have the monoclonal antibody, and also you have the immunomodulatory cytokines. But recently the terms live biotherapeutics been introduced. So what does that mean? So it's actually originated from the probiotics. So initially probiotic, we define that is the any bacteria that give the health benefit if you are taking in the adequate dose. So some of the probiotic, they have specific therapeutic functions and they be used against a specific disease, and that's we call the biotherapeutics. And when we talk the biotherapeutics, it could be the natural isolates or it could be the genetically modified probiotics. When we talk about the genetically modified probiotics, it's actually probiotic with some therapeutic peptides. It's either originated from the microbes or human been implemented, so they do work quite nicely. And in terms of the development of biotherapeutics, it exactly follows the same routes like therapeutics. You have your live biotherapeutics. First, you need to identify, do characterize, either by genome sequencing, and then go for production and clinical trials. So what is the current status of live biotherapeutics? So if you look these slides, you could find that there are live biotherapeutics, those are already been approved and have the huge market value. Most common one used in Australia is the Mutafro, that is derived from the East Asia coli initial 1917. And there are so many others also in the clinical trials. Now, question is that why people should be interested to invest THSTI for live biotherapeutic development. So what are the unique capacity do we have? So THSTI has some unique capacity that hardly you could find any such capacity in any other institute in India. So we have a huge resources of the human associated microbiota. Those are genetically defined. more than 6,000 isolate do we have that we isolated from the multiple part of the country. We have almost like 12 different hospitals that we are collaborating, that's situated on the 7 states and 1 union territory. And those isolates are both from the healthy subjects as well as from the people suffering in different disease condition. To idea to have those isolate from the disease condition to check that if you are giving a specific microbes, whether they do have the pathogen resistance potency or not. So in our labs, when we started this live biotherapeutic research, so we actually adopted 3 different approaches. Use either your microbiome as a therapeutics, where you have a direct clinical samples. Second approach that most of the peoples are now engaged, use microbiota. That means you isolate the microbes and then test whether those microbes could have the therapeutic potency. And third one, we are now recently initiated the bacteriophages, whether those bacteriophages could work against extensive drug-resistant pathogen where antibiotic don't work. In terms of the microbiome, what I can say that we have a major collaborator from All India Institute of Medical Science, Dr. Binit and Dr. Salimar, and All India Institute of Medical Science from the Kolani. Here, actually, our role to analyze the microbiome to identify an ideal donor. So we do analyze bacteria, fungi, and viruses, and then actually we pass that message to the clinician. This could be the most potential ideal donor where you have the maximum efficacy. When we are talking to the microbiota, we have both the approaches. So in the microbiota, we do isolated the natural microbes either from the reproductive tract or from the gastrointestinal tract. And then we tested those microbes against mostly to the non-alcoholic fatty liver disease, inflammatory bowel disease, and also sometimes to the malnutritions. And the second one that actually I feel that we have unique capacity, that is the genetic genome engineering for the genetically modified microorganisms. So what actually we do, once you have your probiotic microbes, they do have some potency, whether you can supplement additional functions. So those functions could be derived either from the humans or from the bacteria or from the bacteriophages. I'm just giving one example here that we have the E. coli initial 1917s. Now we supplemented those E. coli 1917s with the human encoded GLP-1. So these bacteria, they have the capacity to produce and secrete GLP-1 in substantial amount that actually give your health benefit. Same thing we tested for the microbial derived function. So currently we have the 3 molecules. Those actually are produced by certain sets of bacteria. Those are diminished in the disease condition. One is the microbially encoded anti-inflammatory molecules that rhyme from the Helicobacter peugeneyi that are known that their number is reduced in the IBD. Second one is the bile salt hydrolase that is very important to modulate the gastrointestinal tract homeostasis. And third one is the methanolysins. In terms of the phages, we are using even some of the phages functions that has the capacity to lyse the pathogenic bacteria. So this is the major focus we have in the labs. And 2 of my PhD students, they started working on the bacteriophages. We are collecting those bacteriophages from environment as well as the human gastrointestinal tract, and then testing their microbial lytic activity. We have selected extensive drug-resistant Klebsiella pneumoniae, Acinetobacter baumannii, P. aeruginosa, and E. coli, and then we screen. Currently, we have more than 1,000 pure bacteriophages that can lyse or that can kill any sorts of antimicrobial resistance, those gram-negative escape pathogen, and also we have started working on the gastrointestinal tract pathogens. In terms of the wild-type bacteria, just I want to give one example when we are talking that we have the lactobacillus isolate from where those lactobacillus isolate came from. So I'm part of one of the largest cohort, I think, in the Asia of the pregnant women, that is the Garbhini. So in the Garbhini women, this cohort, almost like 12,000 pregnant women been enrolled, and we are collecting their different biological samples, including the high vaginal swab sample. So the lactobacillus isolates that we have, we actually isolated from the reproductive tract of the Indian women. We did the first metagenomic analysis to understand what types of lactobacillus is there. Then we isolated those lactobacillus, those are aerobic or anaerobic, encoded their functions, identified the bacteriocytin or the bacteriolytic functions that can inhibit the pathogens, identified that protein through the LCMS analysis, and then we tested against reproductive tract pathogen as well as the enteric pathogen. So currently we have the 3 Lactobacillus crispatus and 3 Lactobacillus gasseri. They do have the pathogen resistance functions against all the reproductive tract pathogen. In addition, what we observe that these Lactobacillus crispatus, they have fantastic activity against reducing your fat accumulations. And we tested with particularly the mice fed with choline deficient high fat, high fructose diet, and we check longitudinally. If you are giving those mice with this lactobacillus, they reduce visceral fat accumulations as well as the hepatic fat accumulations. This could be one of the potential therapeutics. In addition, this lactobacillus also has potency you could use as a nutraceuticals, because we tested that if you are giving at least 10 to the power 5 to 10 to the power 6 number of inoculum of those lactobacillus, they form nice card within 5 to 6 hours, both in the buffalo milk as well as the cow milk. So in summary, what I can explain that what things right now we do have that we can start working with the company. We have the genetically modified organisms with microbially encoded anti-inflammatory molecules. These molecules we identified from a bacteria that isolated from the healthy Indian population. We optimize these leads, and we have also done the animal study. So till we have the preclinical study. Genetically modified organism with bacteriocins, we have till to the heat levels, and the lactobacillus consortia that we have tested till to the animal levels. With that, I'd like to acknowledge all my collaborators, my funding agency, and I'd like to thank all of you for your kind attention. Thank you.
Thank you, sir, for sharing your expertise with us. I now welcome Dr. Jayanta Bhattacharya, Dean of BRIC TGSTI, to present his insights on the research involving monoclonal antibodies being conducted at our institute.
Good morning to all of you. It's so nice to see again the full house, which normally we don't necessarily used to see. In the interest of time, I'll just walk you through another capabilities that we have at THSTI, which is something is talked about nowadays. Is around how monoclonal antibodies could be used as a precision biotherapeutics. As you know, that antibodies plays very important role in public health. It could be used as immunodiagnostics, immunotherapeutics, vaccines, and also surveillance. And we all know, I don't think this audience, especially from the industry colleagues, it is not new to understand how monoclonal antibodies is playing a very important role along with the vaccines, not only necessarily in the area of cancer, immunotherapy, but very recently we understood how monoclonal antibodies can be useful in tackling infectious disease as well. What I would like to actually highlight here is that, it's, beyond what we do in the lab, how do you actually take some of these leads to actually apply in the public health domain? And what we realize that a public-private partnership is very crucial in not only innovate a product but also we ensure that it reaches to the right population, it is affordable and accessible. And therefore, we believe that proactive alignment between different sectors and stakeholders is essential, very, very essential. What I'll actually show you today is essentially the kind of platform that we have in THSTI, which can essentially start from, as Bhavtosh showed, from a cohort to very rapidly isolate or discover what we call human monoclonal antibodies, which can not only recognize the antigen of your interest, but also can neutralize. And we have the ability to test in preclinical animal model very rapidly. And this is one of those technologies that we use generally, one we call as antigen-specific single B-cell technology platform, as well as very recently we have established what we call as 10x genomics facility, which is essentially very rapidly identify very precisely some of the very powerful monoclonal antibodies, in this case, human monoclonal antibodies. Let me give you an example of how we have used this technology to very rapidly isolate very powerful monoclonal antibodies against SARS-CoV-2 right in the year of 2020, when the Yuan strain was circulating. We actually concentrated a lot on vaccines, but little we realized that India has also developed capabilities in developing monoclonal antibodies against infectious diseases at that point of time. This is just an example. Not only we had isolated human monoclonal antibodies using the cohorts that THSTI had built, but also we have very rapidly tested against their ability to protect against the very virulent Delta strain right here in THSTI, which actually has led us attract some of our friends from industries to actually invest in taking this forward. We also have developed antibodies. It's not about invention of antibodies or discovery of antibodies, but also we realize that you need to also, in order to make it more accessible, more affordable, and more effective, you also need to have platforms that can not only make it more potent or you extend their serum half-life, but you can also do some modification that can essentially make it useful for therapy. For example, it can actually bring in, the ADCC or ADCP effect. That means it can kill the infected cells as well, which is something that we have done in collaborations and improved our ability at THSTI. And very last slide, and we are very proud, and this is a breakthrough which has come from these institutions in working with our another institute at DBT, Rajiv Gandhi Center for Biotechnology, in working with NCDC and Government Medical College, Kozhikode. We know that last year there was an outbreak of Nipah virus, and we thank DBT for extensive support in accessing some of these valuable samples. Because we had developed this capability of actually can, from the samples, rapidly isolate monoclonal antibodies, we now, again, as like COVID, we are the very first institute, not only in India, but in LMICs, we have now been able to isolate very powerful human monoclonal antibodies against Nipah virus, which is something we are essentially now in a position to take forward for clinical development, along with the industry development. And I just sort of highlighted which are the areas where we are capable of doing things at our institutions, which are the areas which we need your involvement, or we need help from the industry working where we can work together, and so that we can actually first develop it. In fact, it is the recent news, CEPI has actually funded about $43 million to actually develop a Nipah monoclonal antibody, which is going to be conducted in India and Bangladesh. And this is where we are, that we have our own monoclonal antibodies, but what we believe is better than what is being thought of taking forward for clinical development, because we have antibodies that can target different antigens. And this is why we should actually, I think we should work hand in hand with industry to first develop these antibodies, which will not only help, cater the patients or the population in India, but also outside India. And this is where I think we should take a leap forward. So when we actually talk about discovery and development, THSTI, and you will hear a lot about this today in different presentations, we have capabilities in running some of these assays that many vaccine manufacturers had utilized during developing their COVID vaccines. We have a GCLP lab, which is recognized by not only CEPI, but also NABL accredited. So we have assays that can, that can do assays in a GLP compliant manner. We have animal house to facilitate preclinical development, and many, many other platforms which can actually support the development. We ensure that the quality is always going to be there when we take some of this invention for clinical development. So this is my final slide, and we often talk about translating innovation into public health impact. And what we need, and therefore, we realized, and THST is a great example of this is that you really need a research platforms, and we have developed a lot of strengths. You need a multidisciplinary team, which we have. In many ways, we have mathematicians starting from biologists to biologists to mathematicians to computational biologists, all of this, and clinical scientists, all of we have here, along with CDSA. What we need to strengthen, we believe, is a public partnership model just to accelerate this process. And we should all be very proud. I think this will make us really proud as an Indian. So what are exactly what we're expecting from industry? I think this is one of the reasons why we're conducting this industry meet today, is that can we discuss how do we actually bridge these gaps which are existing so that we can first develop some of these novel molecules, in this case, monoclonal antibodies, very quickly that can be deployed for public health interventions. As I said, you will hear a lot today from different presentations. What are different facilities that will uniquely find in this campus that you will not, as Amit said, you will not find in other campus, right from the BSL-3, animal BSL-3, the biorepository, the clinicians, the medical research center coming up, and the translator research facility coming up. So I'll stop here. Thank you very much.
Very promising. Thank you, sir, for sharing the insights. Now, no translational research is incomplete without vaccines. So please welcome Dr. Sweetie Samal, Principal Scientist to THSTI, on stage to share the information on the ongoing research on vaccines.
Very good morning, everyone. So I will briefly walk you through the vaccine R&D that we are doing at HSTI. So I believe this slide is very much the industry. It is not new to the industry partner, but we would like to project that in 2029 post-pandemic, the vaccine industry is going to be increased to 94.5 billion USD. And we also acknowledge and understand there are many key players in the global market. So to position the Indian industry, who are the biggest manufacturer in world, and 60% of the vaccine has been supplied, but I will again come back to it. So it is very important to place or put a strong foothold in this vaccine market. There should be a clear-cut understanding of suitable antigen, adjuvant, platform technology, delivery, and the regulatory. And if you look at the vaccine, when it comes to the vaccine, the majority of this vaccine goes for the childhood immunization. However, post-vaccine, there are another area which is coming up, which is the adult vaccination, and the preventive vaccine, therapeutic vaccine, and another crucial area is the travel vaccine. So these are the three areas which are also going to put a very strong space in the vaccine market. So we are ready as a vaccine industry to show our mark in the global industry. So post-pandemic and during the pandemic, we work very closely with the industry partner, and that helps us to identify the key research gaps. Why the vaccine, even though we are the biggest manufacturer, but most of this vaccine technology has been borrowed from West side, and why in India we are not able to provide not only this technology, but other research resources to the industry. So these are the major research gaps that identified as the indigenous novel vaccine platform or technology, the preclinical animal models, which is one of the major prerequisites for any vaccine development, understanding on the pathogen diversity and the emerging infectious diseases, and some of the technological and methodological gaps, advanced research tools such as AI, structure, virus-specific assays, and some of the knowledge gaps, immune response mechanism, long-term efficacy. Because for some of the pathogen, as we have also seen for the COVID, I mean, we knew that this RNA virus is going to change a lot, and the new the vaccine which has been uh, developed using the Wuhan strain may not be that much efficacy for the new Neogen variants. So to bridge the gaps, it's very much necessary that not only in the manufacturing or development part, but from the fundamental part also we come close and build a public-private partnership. So why THSTI? So it's not during the pandemic. THSTI, we all should acknowledge and believe, is the only institute in India which has played a key important role for the indigenous vaccine development, and India's first indigenous developed vaccine for rotavirus is the Rotavac, which has given the regulatory approval in March 9, 2015. So Teachers Day, from the inception of this institute since 2009, is actively working with the industry, and being the academic research institute, they understand the industry language. So that's why maybe post-pandemic, we gave a very serious thought that we should come up as an academic institute from India to bridge that gap and augment the vaccine R&D at HSTI. So the aim is to facilitate and actively engage in developing novel and cost-efficient vaccines. Because if you look at 80% of the population belongs to the LMIC country, and during the COVID, it also come to notice that this vaccine accessibility is not available to each and every individual across the globe. And cannot be achieved having a basic translational basic fundamental facility in the institute. That also being acknowledged in THSTA, for which we are now developing some specialized academic facilities like Vaccine Design and Development Center, which understand the industry needs. We are coming up with translation research facility and medical research facility, which you will hear more. And now we have enhanced the engagement actively with the industry and startups, not to only have a collaborative R&D, but to understand what is the gap, what industry really wants, and what academia research scientists they require from the industry. And definitely to do all this, we need the funds. And as said rightly by our director in the beginning, we cannot only dependent on the government funding or the public funding, but however, we can also look across the global fundings, and jointly, we can also secure those funding for this vaccine R&D. So this is our 12 years pathway, which we have achieved in the vaccine development journey. And we have a very strong knowledge on the lead identification. Unless and until you know the antigen, the vaccine cannot be developed. Everybody says that the COVID vaccine has been achieved in 300 days, but the research has been done for the last 15 years on the coronavirus and SARS-CoV-2, which have identified the spike protein is the antigen which can give you full protection. Then come to the lead optimization. We as a scientist are very good in the lead optimization. Not every antigen can give you full protection. So that is also one of our strengths. Then come the preclinical POC. For every vaccine to go to the human clinical trial, you need a preclinical model, and viruses are very notorious. For each and every virus, you don't have a preclinical model, and also there is a BSL-3 and BSL-4 regulation. So that we are trying to minimize this. Another term that we understand is the process development. So if you ask any academic or research scientist what is the DP or DS means, we never had that clue. For us, developing an antigen and then giving it to a preclinical model, getting a full protective efficacy, has done our work and publishing in a high research quality paper. But with working with the industry, we understand that process development plays a very key important role for any product development, and that we are working currently with a stringent QC and QP policy. And then IP, patent. No industry will come forward to us if we don't have a patent of our product. That also we have acknowledged, and we are moving forward to have our patent or IP protected as soon as possible so that we can enter into the global market. And beyond that, this is our future vision that we should have a pilot scale GMP manufacture. So we demonstrate your product has already gone so far. So the industry doesn't have to again start from the beginning, which takes a big big setback for the industry. And also we would like to demonstrate the phase 1 clinical trial in academic setup so that industry can quickly take this product to the market. So the most sought after viruses, these are the virus panel that we are working on, where more than 50 faculty and scientists and more than 60 research technical personnel and PhD is working. And as an academic scientist, our keen interest in the basic and fundamental research, but post-pandemic, we are trying to exploit this fundamental research in some of the translational and product technology. So these are our key expertise and resources and capacity. We can do the antigen designing, and we can demonstrate the suitability of the vaccine platform. For every virus, you cannot use the same platform, whether it's a mRNA, inactivated or protein based, or the like virus vector. So a different vaccine platform is needed for different pathogen. We also have developed few novel indigenous vaccine platforms. We are developing the disease-specific preclinical animal models up to BSL-3. We have virus and vaccine-specific assays ranging from molecular, virology, serological, immunoassay, immunoprofiling, and omics, which can give you a value to down-select the promising candidates. Now we have a scale-up facility for the protein antigen up to 5-liter fermenter, which we would like to take further up to 20-liter. We can do a comprehensive analysis of biochemical, biophysical, and analytical characterization of the antigens. And definitely we have generated the reagents. Maybe for the industry, it is not, cost doesn't matter. For example, if you want a NEPA polyclonal sera to test some of your vaccine or monoclonals or antiviral, we have this and we can provide it. And we are also coming with a virus banking in coming years. So coming to the platform, this is one indigenous multivalent vaccine platform which has been developed at THSTI post-COVID. So in 2021, it has been started in a scratcher paper. We received the patent in January 2021, Indian patent. Now it has been applied to various countries for the PCT. So this is a plug and play platform where it has the flexibility to display multiple antigens from the same virus or the different virus. As a virologist, I always say we are lucky to have SARS-CoV. It is the most easiest virus we have worked through. But if you talk about the influenza, monkeypox, or other viruses, the viruses has this complexity, or dengue viruses. It has different serotypes. The viruses changes very quickly. And these are the hazards that put a vaccine industry to invest lot in the R&D. So this platform is a plug and play platform. Different antigens can be sampled. And we have the POC established in the bacterial expression system. I think every industry has this bacterial system established. Another thing is the structure AI-driven native soluble antigen and BLP platform. We can produce the native-like antigen of the virus structure protein. We also have a VLP platform that has been demonstrated for the for the hepatitis E virus, and we have another platform which is a bivalent circular mRNA vaccine platform against SARS-CoV-2. So these are some of the vaccine portfolio that has been developed post-COVID within the 3 years, and we would like to mention that we also value the industry timelines, and you can see the robust way. Now we are working to make the product coming up. For the phase 2, we are coming up with 2 facilities. One is the translational research facility, where we will like to develop and pilot scale GMP production for the protein-based vaccine and for the viruses up to BSL-2. So those viruses can be used further for the live attenuated or inactivated virus or for the controlled human infection. There another facility is coming up for the medical research center, where we will be building a center for controlled human infection, and that can decrease. It will be the first in kind in most Asian countries, and it can reduce the cost of the vaccine for the clinical trials drastically. So why you are doing so, I don't want to have the repeat, but as a research scientist from the public academia institute, our focus is to have this for the target is the developing and the least developed countries, so that we can have a cost-effective vaccine for everyone, self-reliance, and we can have a rapid response to the emerging viruses. So with this, I hope this the first get-together at HSTI will enable us to connect, collaborate, and co-create to accelerate indigenous vaccine R&D. With this, I will stop, and we'll be here for the whole day for you. If you have any questions, please feel free to interact with your scientists. Thank you.
Thank you, ma'am, for sharing your engaging insights with us on vaccines. Please join me in welcoming Dr. Dinesh Mahajan, Associate Professor at THSTI, to acquaint us with the current developments on the application of small molecules, focusing on TB.
Good morning, everyone. Before going into project-specific deliberations, which is more focused on tuberculosis, I will give you some insight on what kind of efforts we have at THSTI, specifically on drug discovery. So if you see THSTI as an institute, so we are working on different kind of diseases, particularly diseases which are of national interest. And if you, see more deeply, so there are different kind of disease research groups, either working on disease pathogenesis or disease pathophysiology, using clinical samples or developing animal models and employing molecular and omics analysis. In the virtue of this, they are also identifying different kind of drug targets, and some of them are also screening libraries and identifying different kind of hit molecules to facilitate drug discovery efforts. And interestingly and very useful, we also have CDSA. CDSA is a clinical development service agency, which is a department within THSTI, and having expertise on doing different kind of clinical trials ranging from phase 1 to phase 3 clinical studies. So this is a set stage to do drug discovery. And complementing to this, we have a center which is disease agonist, having expertise ranging from computational based biophysics, computational based bioinformatics. We also have synthetic organic chemistry, which can facilitate structure activity relationship while synthesizing new molecules. And we also have drug metabolism and pharmacokinetic analysis facility at THSTI. So all these interdisciplinary skills and infrastructure are working together to create a synergistic environment, which is leading to a very healthy and robust infrastructure as well as intellectual capability at THSTI. And the aim here is we work on specific disease, create new drug targets, generate new molecules, and progress those molecules on drug discovery pathway, and come to a stage where we have a drug candidate ready for IND enabling studies, and then join hand with the industry and do those IND enabling studies, and then come back and use CDSA as a center to do clinical trials. So if you want to know more about this center and drug discovery capabilities, I will appreciate you join us during breakout session on drug discovery at 3rd floor of THSTI. So coming back to this program, which we have on tuberculosis infection. So we started this program like 2 or 3 years back. So there is a clinical need, and this is, this is a long pending issue in TB drug discovery. So we need to shorten the treatment, or we need to improve the treatment of drug-resistant TB. We know that MDR and XDR, the success rate is low and the treatment is very complex. So based on this understanding, we created a kind of broad target product profile and started a discovery program. The idea was, if you have a molecule which can address either of these 3 objectives, and additionally that molecule should be compatible with existing TB drugs, then clearly you have a clinical and business case for tuberculosis drug discovery. So based on this thought process, so our TB research group led by Raman, Nishit, Amit, and Krish, they actually are doing those efforts to identify different kind of hits using these 3 different approaches. I will not go into detail, but so the approaches that they are using is either based on target or phenotypic screening or host understanding, how the host pathogen interactions are playing role in the pathogenesis of the disease. So I will deliberate on one specific project that is a culmination of the work done by Dr. Ramandeep. So where he did a large library screening based using phenotypic assays, and he identified actually a pool of hit molecules having differential activity against drug-sensitive tuberculosis. So based on MedChem understanding and based on the therapeutic profile that we thought in the start of the project, we selected one hit molecule with a decent activity, with a decent structure, drug-like properties, and we started evaluating that molecules on the path of discovery. We saw that this molecule actually demonstrated a very nice dose response both on in vitro assays and also on intracellular MTB infection. So with this understanding, we venture into investigation to understand the mechanism of action, and these studies reveal that there is a particular host and pathogen enzyme, amidase C, which is a hydrolytic enzyme, which is required for the bioactivation of this molecule. And part of this work we published recently in a publication. So with this understanding, we define a kind of workflow, keeping in mind the target product profile, and based on that, we define our primary assays, secondary assays, and we did a 4-tier of SARs to have a better understanding about the pharmacophore of this molecule. And this effort led to a series of molecules. And I will tell you more about 514 and 536. But eventually we end up with a novel series. This series is protected as an Indian patent with THSTI. So we are quite close what INH is doing either in preclinical models, both in vitro and in vivo. And these molecules are also active against MDR and XDR clinical sales that you will see in my subsequent slides. So this is one of the lead, DR514. So this lead is not only active against RV strain, but it is also active against lab-generated CATG mutant bacteria. So this data is of infected macrophages with mutant CATG. So you can see the last two bars. The second last is INH, and the last bar is DR514. So definitely this molecule is active against drug-resistant tuberculosis. We also evaluated this molecule using different clinical strains, and we found equipotency for this molecule on clinical strains, whether it's MDR or XDR. So this was exciting. So we also did a kind of checkerboard analysis to see whether this molecule is not antagonizing or interfering with the activity of the existing drug. And we are quite safe. There is no interference. And we also evaluated this molecule in 4-week animal efficacy model, and we saw a decent, significant activity. Compound 30 is actually compound 514, and we published this data. So this is another lead, 536, which is a second generation to 514. And you saw this standalone treatment of 536 and 537, which is very close to what INH is doing in animal efficacy. We also dose this molecule in combination with the existing first-line treatment comprising of isoniazid, RIF, pyrazinamide, and ethambutol. And there is there is a marginal better effect if you compared with the, we replace actually INH with our molecule, the 536. So we replace H with 536 in the combination arm, and we observe some better effect in lung, but a significant change in the liver spleen infection. So with this, I will conclude that we have a novel series, and we demonstrated its activity both in drug sensitive as well as drug resistant tuberculosis strain. Is orally bioavailable, and animal efficacy is also established. The synthetic route is amenable for any large scale synthesis. So right now we are working on DMPK profiling and also working on drug-drug interaction based on pharmacokinetic analysis. We would love to collaborate for further development on this molecule. If you want to more or understand more on this project, please do talk to me or Ramandeep. So this is acknowledgement. Thank you. I'm open for any question if you have during the breakup session. Thank you.
Thank you, sir, for the informative talk. Right treatment require right diagnosis. I now invite Dr. Gaurav Batra, Associate Professor, THSTI, on stage to discuss about the ongoing work on in vitro diagnostics.
Good morning, everyone. So what I'm going to do is to give you some highlight or share some highlights on the work we are doing on in vitro diagnostic development at THSTI. So I'll share what we are doing here at THSTI on innovative diagnostic product development. So we use different technology platform to reach to a final product that includes generation of monoclonal antibodies. We use different platform, hybridoma, phage display. We make recombinant antigens and antibodies, including optimizing the production of antibodies for diagnostic product development in 3 different expression systems. We utilize antibodies for the detection of antigens and antigens for the detection of antibody in the body fluid, and develop assays both in well format as well as point of care lateral flow format. Apart from immunoassays, we also develop molecular assays, both in traditional RT-PCR format as well as CRISPR-based nucleic acid detection platform. And we apply these platforms for diagnosing tropical febrile illnesses from dengue, malaria, now Zika and chikungunya, and antimicrobial resistant, fungal infection, and recently started working on early cancer detection. One of our aim is to enhance the performance of point of care test to bring it to the similar level, at least closer to the central lab test, so we can bridge this important gap. For that, we use traditional rapid test as well as unique nanoparticle-based fluorescent assays, which allow us to enhance the sensitivity of assay in some cases more than 50 times. This is just one example where, based on the target product profile provided to us by FIND and WHO, we were able to make a malaria test which is 50 times more sensitive than the WHO pre-qualified visually read test, and it has utility in the elimination settings for malaria. For dengue, there is a problem that in the secondary dengue, the NS1 antigen detection test does not perform well because of the immune complex formation. And the commercial assays have problems of serotype level bias, means these tests do not perform well for all the 4 serotypes. They perform well for few serotypes like 1, 2, or 3, but not 4 and other combinations. So what we were able to do, we generated a very large repertoire of monoclonal antibodies, close to 100 antibodies, extensive analysis, and we were able to find some of the antibody in sub-picomolar affinity. And we were able to use these antibodies to develop an assay which work equally well in the secondary dengue, not just in the primary dengue. And even a rapid test performed better than the market leading ELISA for the detection of NS1, and that too without any serotype level bias. Additionally, this test does not cross-react with Zika or other flavivirus NS1 antigens. During COVID time, we also developed large repertoire of monoclonal antibodies against SARS-CoV-2 nucleocapsid protein, develop assay both in traditional rapid test as well as a fluorescent upconverting phosphor nanoparticle-based test. And we benchmarked it against SD biosensor, which at that time was the best product in the market. And what we found, the test which we developed in a visual readout format was similar to the SD assay, but the assay which we developed using the upconverting phosphor nanoparticle, that was even at CT value of 35, which means very low level of viremia. We were 20-fold, having 20-fold better sensitivity than any colloidal gold-based assay, showing the power of this upconverting phosphor nanoparticle coupled with right kind of antibodies. Again, during COVID time, we developed battery of assay for the detection of antibodies against SARS-CoV-2, targeting RBD as well as nucleocapsid protein, develop IgG, IgA, IgM ELISAs, including quantitative ELISA for different variants, including Delta and Omicron and sublineages. And this was extensively used for different serosurvey studies, as well as to assess the ability to quantitate what is happening on ground. This is a comparison between our assays and commercial assays from Abbott. There is hardly any difference in the performance. This data shows that we were able to show that during 2022 early Omicron wave, we were able to see that there were extensive asymptomatic infection happen, and we could able to quantitate this. and this data went to government to make appropriate vaccine booster policy. My colleague Neeraj, he works on AMR diagnostics. So he's working on how we can reduce the time for pathogen culture, particularly the escape pathogens. And he's working on device which allow this type of enrichment. So the sample can be, the culture can happen in 2 hours time. He's also developed multiplex PCR assay for escape pathogens. Another colleague of mine, Dr. Nishit, he's using CRISPR for detection of different drug resistance pattern in AM tuberculosis. So basically our key capability at THSTI is immunoassay development as well as robust molecular assays. And with this, I would like to thank all the industry, particularly industry representative and all the dignitaries and other guests. And we are open for discussion, particularly into the breakout session. Thank you.
Thank you, sir, for giving us a detailed overview and sharing your insights. Please join me in welcoming Dr. Amit Awasthi, Senior Professor at THSTI, on the stage to share his insights on the latest research going on in the field of cell therapy at our institute.
Thank you. So I will be briefly talked about the work what we are doing in our laboratory on cell therapy. So basically, I mean, we know that cell therapy market is actually now across the world is booming, and particularly for the chronic diseases, it is something which is now being used. For cancer treatment, the CAR T cell therapy for the liquid tumors has been, approved in the US as well as even in India. And now for other autoimmune diseases, for which normally the monoclonal antibodies were being used, now cell therapy has shown some promise, and these data has been, published in the domain. So you can see the market across the globe is actually projected to increase significantly, and from like 2031, it's going to be jump 52%. And similarly, India, the growth of the cell therapy is also booming. There's a lot of partners, actually, industry partners, startups actually working on the cell therapy product after the approval of the first CAR T cell therapy, which came out from the Mumbai Immuno Act in partnership with the Tata Act. So we know the cell therapy can be broadly divided into two categories. One is the immunotherapy and one is the non-immune cell therapy, which is the stem cell therapy. In case of immunotherapy, you can have, CAR T cells, which is one of the therapy which has shown promise in cancer and other autoimmune diseases treatment. And then other than CAR T cells, you have non-CAR T cell therapy, which is not yet in the market, but actually initial results shown promise, and people are working on the off-the-cell therapy using NK cell, NK CAR, and so on and so on. When you have B-cell lymphoma, you can really target B-cell lymphoma through the CAR T cell because the T cell can kill B cells. But when you have T-cell lymphoma, the T cell cannot kill T cells because T cell can also be self-detrimental. So in that case, the B cell is probably a good target. And I know some of the DVT institution is working on the B-cell CAR therapy to target T-cell lymphoma. Then we have other therapy other than the macrophage. People are shown these are in the academic settings, not yet actually came out to the market. The advantage of CAR T cell therapy is they are very precise. This is again comes in the precision medicine. So if you really actually activate the T cells or immune system in a manner, you can really target, the cancer cells, and this remission can be long term. So this is very precision driven medicine. The other thing is after the identification of different CARs, you have different generation of CAR T cells. And each of these different generation of CAR T cell to decrease the cytotoxicity, you can modify the cytoplasmic domain of the CAR to have lesser cytotoxic effect. You can keep adding different kind of permutation combination of the cytokine domains or the other T cell activation domain in the cytoplasmic domain, so that actually you can very precisely kill the target. And this is this is how all started. Emily was the first patient in the world have actually got the CAR T cell treatment when she was actually 5-year-old in 2010. And then she celebrated cancer-free 11-year last year, and she is complete remission. So this is the power of cell therapy. This is the precision, lesser side effect, and it can be long lasting because ultimately immune cells, which you actually put in the patient, can also generate memory response. And every time you have these cancer antigen comes up, the memory response of T cells can actually start working again. So this is a powerful tool what has been actually, and this is just a kind of that's how it works. This is like cell therapy product. You have to take out the patient's blood, you isolate, cells, PBMCs, you engineer them in the lab, in the Petri plate with the different vectors, different lentiviral based vector, and then you infuse them back into the patient and then monitor the patient for the longer term. This is the kind of landscape of CAR T cell therapy or cell-based therapy. You can see started 1992, how to engineer the T cells, how to modify the T cells. The effort was kind of very hard because it was very difficult because immune system is a double-edged sword. So if you tweak it in a way, it can be beneficial, but if you tweak it in the wrong way, it can be detrimental because same immune system can induce autoimmune disease, inflammation, and the same immune cells can kill the tumor cells. So it took longer time to bring the CAR T cell therapy in the public domain with multiple case reports. And now you have multiple therapy has been approved in the US, as well as one therapy has been approved in India. So what we have, what is our capability? So the idea to use CAR T cell, because our lab is working last so many years. In fact, my experience in the T cell domain is last 20 years. So I am only working on the T cell, whether it's a vaccine, whether it's a cancer, whether it's other autoimmune disease. So that actually bring us together the expertise what we have generated a lot of models and the tools and the assays within the lab to use this system and platform to cater and bring industry together. Can we take it to the next level? So these are the different platforms what we have generated. We tested in the preclinical settings, and the clinical partners we are talking to different partners, and AIIMS Delhi we have oncology department. We have collaboration with AIIMS to see how we can take it forward. In addition to this, we have assays validate CAR T cells both in vitro and preclinical model. And then the laboratory has actually developed its own vector, which will be tested in the settings. One of the important thing of CAR T cell therapy, once you have the vector, which is kind of validated, is giving really good binding to the antigen. The other thing, how to put this vector back into the T cells or in the immune cells. So one of the thing is the lentiviral system, which is very efficient to infect T cells with this CAR vectors. And currently we have like different generation of lentivirals, second generation, the third generation. And our lab has successfully tested both the second generation and the third generation lentiviral vector. And you can clearly see the efficiency of third generation and the second generation and the third generation lentiviral vector can put the CAR construct in the T cells. As you can see on the graph that percentage almost 80%. And then we tested different permutation of combination of these lentiviral vector, and we achieved most of the difficult tasks in the lab to have the concentrated lentiviral vector at least in a billions, right? So one small microliter of lentivirus containing your CAR T cell construct can infect a lot of T cells, and these infected T cells will go back to the patient. And this is what we have shown, that if we generated third generation lentiviral vector having CAR construct into it, and you can clearly see that small amount, even 1 microliter of the lentivirus is sufficient to infect transduction efficiency up to 60%, which is remarkable. And then we have tested this functionality of this CAR T cell, what we have packed in the lentivirus in the in vitro killing assay, and you can clearly see the transduced PBMC, which is green in the color, having CD19 antigen into it, and the CD19 expressing CAR T cells is able to kill your B cell, which is in the in vitro killing assay. as compared to control and untransduced PBMC. So with this, now we are having a proof of concept. We are ready for this, kind of proof of concept and the various assay what we have developed in the lab. And now we are in a stage that we would like to have the GMP facility for the CAR T cell or cell therapy, where we can cater the NCR region. Right now, there is only 1 or 2 facility, which is not sufficient to cater the larger need, because ultimately the cell therapy will not only important for cancer, but it ultimately will also important for autoimmune diseases. And the greater result has been shown as published in the NEJM recently, where the B cell mediated autoimmune diseases has been targeted, and these patients are having remission at least 2 years now. So this is a kind of great we are starting, and we will be happy to collaborate in the breakout session. I will be happy to answer any question related to cell therapy and how we can partner using our lentivirus platform, what we established, or the various other platform regarding cell therapy. Thank you very much.
Thank you, sir. Very informative indeed. For our next speaker, please welcome Dr. Ramandeep Singh, Senior Professor, THSTI, to kindly walk us through the various facilities present at THSTI.
So, good morning, everybody. So today I would be going through the strengths of the various facilities which are developed at THSTI. So at THSTI, we have developed 9 different facilities, and all these facilities have state-of-the-art infrastructure and has trained manpower to generate high-quality data to support clinical and preclinical research in disease areas that have major public health implications. So the first facility that I would be talking about is the bioassay laboratory, which was also mentioned briefly by Dr. Janta. It is designed as per NABL guidelines to support development of vaccines and various therapeutics. It is the only lab that is part of CEPI globalized network for facilitating COVID-19 vaccine development. The facility has trained manpower and a robust quality management system to generate data from various cellular, biological, and molecular assays for various viruses, such as SARS-CoV-2, flaviviruses, and influenza viruses. This bioassay laboratory serves as a platform to collaborate with various national and international organizations to facilitate product development that is related to either vaccines or therapeutics. The facility had collaborated with various international and national organizations to facilitate product development for COVID-19, and out of these collaborations, 2 vaccines, Corbevax and HSP GPOVAC, were eventually approved for clinical use. The second facility is the biorepository facility, which has been funded by DBT. It is the largest and most varied research bioresource center in India. It is compliant with ISO 20387 compliance. It has state-of-the-art infrastructure, and it archives more than 1.6 million biospecimen aliquots, which have been arranged from disease and various population-based cohorts. Among the services which is provided by biorepository is for biospecimen archiving, processing. It also provides workshop training to people for processing biospecimens and to tell them about biorepository guidelines. It also has a well-structured access policy where a request for samples is received and reviewed by a control committee before the samples are shared with academics and industries. The biorepository facility has supported 19 industries and 58 academic institutes with COVID-19 resources, and have also participated in validation of 8 diagnostic tests. From the bioresources available, they have also generated panels for preterm birth, and they have also generated sera and plasma plasma pellets for serosurveillance of dengue, chikungunya, and COVID-19 virus. It is also part of ISBER. Experimental animal facility has been recently renovated and is a SCA registered facility. It has the provision of housing mice, guinea pigs, rats, and hamsters. The maximum capacity of the facility is around 55,000 mice. It has dedicated suit areas to work with both immunocompetent and immunocompromised mice, and it also has dedicated area for histopathology and transgenic laboratories. In this facility, we have generated various animal models to study basic and translational research in communicable and non-communicable diseases. We also have a ferret facility in the campus, which is the first ferret facility in the country. It has separate breeding, housing, and experimental room. It is BMS monitored. And among the various services which is provided by the experimental animal facility, these include toxicology studies, efficacy studies, cryopreservation services, and histopathology services. The facility is in the process of filing accreditation from AAA LAC. At THSTI, we also have a biosafety level 3 facility. It is an RCGM approved facility which has 2 dedicated lab areas for working with viruses and bacterial diseases. Each module has a dedicated culture area, animal holding area, and a procedure room area. And this facility also has a common instrumentation room and an automated BMS. In response to the pandemic, we had developed a PRNT-based and CPA-based assays. And these assays were used to analyze clinical samples which were coming from either serosurveillance studies or clinical trial studies. And using these assays, we have supported several industries and academics for product development, clinical trials, and serosurveillance studies. And using these results, 5 vaccine candidates were eventually given a DCGI approval for their clinical use. At THSTI, we were the first one to develop animal models, which was hamster model and transgenic ACE2 model to study SARS-CoV-2. This model had been used to study disease pathogenesis, immunopathogenesis of SARS-CoV-2, and this model had also supported several industries and academics for evaluation of their vaccines and drug candidates against SARS-CoV-2. We have evaluated more than 25 vaccine candidates and more than 30 therapeutic drug candidates for SARS-CoV-2 using these animal models. In addition to the animal models, Immunology Core Laboratory also has the expertise and state-of-the-art infrastructure to study antigen-specific T-cell immune response in clinical samples. The lab has generated various T-cell assays such as ELISpot assays, cytokine quantification assays, T-cell proliferation assays, as well as intracellular cytokine staining assays. Using these assays, various clinical samples were analyzed for their T-cell based immune responses, and this eventually led to the approvals of several vaccine candidates such as Sputnik 5, Sputnik Night, and Corbevax for clinical use after DCGI approval. We also have a data management and data science center at THSTI, which provides data management support for intramural and extramural clinical studies. The center has a diverse portfolio with 14 ongoing and 10 completed projects. The center has also automated several processes. They developed technologies to automate processes for their clinical studies. They've also developed certain AI-based technologies such as GOG to estimate gestational age and also an algorithm to identify fetal parts. They've also designed platforms such as T-mean where the data can be remotely accessed. And the center is compliant with global centers and also works in partnership with various national and international organizations. Another center at THSTI, which Sweetie was mentioning, is the Vaccine Design and Development Center. This center has been established to accelerate vaccine design and development by bridging the gaps between industry and academia. The center has the capability to design next generation subunit vaccines. It also has the capability to purify recombinant proteins from either bacterial or mammalian cell culture and to biochemically and biophysically characterize the purified material. Using an indigenous platform, which is a nanoparticle-based platform, the center can design multivalent subunit vaccines, which can be evaluated in various preclinical animal models. The center also has the capability to purify material from 5-liter cultures, and they are in the process of filing accreditation as per ISO 9001 guidelines. This is the technology readiness levels for various vaccine candidates, which are being designed at THSTI. SARS-CoV-2 beta coronavirus and Mpox vaccine virus and influenza virus, they have given efficacy and proof of concept studies, and THSTI is in talk with industries to take this forward. At THSTI, another center we have developed is multi-omics facility, where the idea is to generate high quality data for genomics, proteomics, and lipidomics. The facility has state-of-the-art instruments to do either sequencing. In sequencing, we can do whole genome sequencing, 16S ribosomal sequencing, or RNA sequencing. We also have the instruments to do label-based and label-free protein identification and quantification. We also have HRMS, GCMS, and orbital fusion platforms to identify and quantify various metabolites, and protocols have also been standardized to do lipidomic studies. So this is a summary slide where I have highlighted the strengths for every facility. And these are the facility coordinators. We would be happy to talk to industry and others during the breakout sessions. I would also like to acknowledge our past and present ED, Dr. Karthikeyan, for their guidance and their support in running these facilities. And we also acknowledge the funding, generous funding received from Department of Biotechnology through translational research programs and INSEP funding mechanism. And these facilities had contributed significantly to the 150 publications, 10 patents, and 6 tech transfers from the institute. Thank you.
Thank you, sir, for providing us a detailed overview of the myriad facilities present at THSTI. Now, please join me in welcoming Dr. Nitya Vadhva, Senior Professor at THSTI, to kindly brief the gathering about Clinical Development Services Agency, CDSA. Thank you.
Good morning to the esteemed guests and all my colleagues. I would like to extend my heartfelt gratitude to everyone for joining us today for this SYNCHN meeting. And I will be providing an overview of and some insights into the Clinical Development Services Agency. CDSA, as it is called, is a center within the BRIC THSTI, and it has a mandate to support and nurture clinical product development and clinical research capacity in the country. It fills a unique niche in the publicly funded landscape as being the only publicly funded agency in the country. And CDSA operates through 3 functional verticals. One is an academic clinical research organization where we have capabilities of providing comprehensive end-to-end clinical study support to investigators, sponsors, and the industry. Then we have a training school where the aim is to build a skilled workforce capable of undertaking complex clinical studies. And then we have a research support and resource center, which fosters and fortifies the clinical research environment through strategic initiatives, which I'll be talking about shortly. Now, in order for CDSA to fulfill the objectives of these functional verticals, we have multidisciplinary teams of highly skilled professionals who ensure seamless and efficient support across all the areas of operation that are there. And so we have a clinical and regulatory science vertical, we have a clinical portfolio management, IT and data science, biostatistics training, and then of course the administration and finance support services without which we could not function. In addition to securing proficient manpower, we have strengthened our clinical trial support capabilities by, establishing a very robust IT infrastructure. So we have validated and globally compliant platforms like the clinical trial management system, clinical data management system, electronic trial master files, a biorepository management system, and several other processes and mechanisms to support the clinical trial activities that are there. Besides that, we've also standardized processes, and we have a constant endeavor to advance the quality of project management, data management, and trial support that we provide. The academic clinical research organization, so all the activities of the academic clinical research organization to undertake and provide end-to-end study support is contributed by the multidisciplinary teams that we have, and we have capabilities of providing support across all the stages of a clinical trial life cycle, from planning to setup to conduct, analysis, report writing, and publication. And the multidisciplinary teams are working cohesively to deliver high quality research. The clinical portfolio management activities again encompass, so I'll be talking about now the capabilities and strength of the different teams that are there. So the clinical portfolio management activities encompass again the entire life cycle of a clinical trial, right from the feasibility to site selection, site initiation, participant recruitment, and then site closeouts, project management, and very thorough monitoring, which includes central and on-site as well as remote monitoring. The clinical science offers proficiency in developing robust protocols, designing comprehensive studies, securing grants, preparing meticulous study documentation, and coordinating seamless execution of clinical trials and studies. Another important component of the clinical science or contribution or strengths are the safety management and medical monitoring that we do. The data science unit of course, does database designing and data coordination for for large multi-country, multi-center studies. And we have different databases and approaches that we use, which are validated and compliant, and they could be commercial data management platforms or open source data management systems, depending on the need of the study. And we also have capability and demonstrated expertise in developing bespoke data management systems. And the data management support that we provide could be paper-based or electronic CRF-based, depending on the requirement of the study again. The biostatistics unit, besides providing professional statistical consultancy with comprehensive multilingual programming support. So besides providing the regular support on supporting the writing of the proposal protocol, the report, developing the statistical analysis plan, doing the statistical analysis, and contributing to the manuscript. They also support the other teams within CDSA for real-time data monitoring, automation of reports. Besides that, we have capabilities or strengths for development and application of advanced statistical methods. An exemplar being that we developed our package for sample size estimation for clinical prediction. And we have capabilities in adaptive trial design, in longitudinal data analysis, prediction modeling, machine learning, and Bayesian statistics. So this is just to give you a flavor of our portfolio of projects. We have 40 plus projects, which could be at different stages. Some are completed, some are ongoing, some are just starting, and they are varying risk, number of sites and participants across a variety of disease areas. So we've done regulatory trials, one of them being, we've just completed a vaccine trial, which was done across 18 sites with enrolling 12,000 participants. And then we've also contributed to the large longitudinal studies where there's long-term follow-up, and from where the discovery research of THSTI also is, the data is generated. We also contribute to the national nutrition surveys that have happened, one being the comprehensive national nutrition survey, and the very recently the diet and behavior survey studies that are happening. So the training school. Now, the training school builds research capacity through high quality training. So traditionally, we do short-term training programs, which could be in-person or online courses as well, on different aspects of research, like clinical research methodology, good clinical practice, good clinical laboratory practice, ethics in biomedical research, even the current regulations like the NDCT 2090 rules that are there. And we've conducted more than 200 trainings across different institutions and have a national footprint. We've also provided training to the SAARC countries. And another important component of the training school is education. So we've started a master's in clinical research with specialization in clinical trials. And the degrees awarded by the Regional Center for Biotechnology. The USP of this master's program that we conduct is that we provide hands-on clinical. There are not just didactic lectures. Of course, besides the case studies and all, we provide hands-on clinical trials exposure. The students have a 3-week internship where they are exposed or immersed into clinical trials besides having a project dissertation. The other initiatives that we've taken to strengthen the clinical research landscape in the countries, one is we have developed and launched a clinical trials toolkit, which is an interactive route map and practical guide for researchers in India, particularly those who are, it was developed for those who are going to be doing clinical trials in investigational medical products, but any researcher can use this resource that's available. It provides information on the best practices and the current legal and practical requirements that are there for conducting a clinical trial in India. We have an electronic ethics committee, which is an online workflow management software for ethics committees. So it helps the ethics committees to, function, to track their submissions that are there, generate queries within a secure environment. And the documents can also be saved within a secure environment. We've integrated the Common Ethics Committee application that was developed by ICMR in this software. Another initiative of ours is the GCP Professional Certification Scheme, which is a competence standard of GCP professionals using internationally accepted best practice for assessment and certification. And this is based on the ISO 17024. Besides that, we've also contributed to the regulatory science and policy support space, where we have contributed to guidelines on nanopharmaceuticals released by CDSCO. We've developed vaccine manufacturing landscapes of India for DBT. We've done consultations to understand the barriers and enablers for experimental research in healthy human volunteers. We've also contributed to the bioregional guidance on human infection models and done a consultation with regulators in preparation for the CHIM studies before the ICMR guidance came out. We are a WHO collaborating center on clinical and translational research for innovation and access to medical products. And with all the strengths and capabilities that we have, we are now developing a program on regulatory affairs and regulatory science. These are some of our collaborations. Thank you.
Before we go any further, I'd like to extend my welcome to Dr. Rajesh Gokhale, Secretary DBT. Welcome. Before these talks fade away, if there are any questions that you want to ask the investigators, we can take those. The industry speak session is still there, but you can still interact now briefly before we go on to Dr. Devan's talk. Just hand over the mics to people.
So the question is, does TSTI has the capability to do developmental and reproductive toxicological studies? And this is becoming mandatory now for any vaccines that could be administered to women subjects or pregnant subjects. So any capability that exists, would love to know that.
We don't have any GLP accredited lab such, but we can establish protocols and can do exploratory toxicology studies very quickly since we have all the element in place.
Hi, thank you so much. It's a wonderful session so far. My question is regarding chimp studies. I think it's very important aspect of vaccine development because now we know that we don't have any epidemics, pandemics, and efficacy trials are really very challenging to do at different countries. And only the controlled human infection models are important, which have been happening in European countries. So it's very exciting area, and I would like to know what are the, status of this, and when we are going to have this excellent initiative from this team. Thank you so much. Hi, Bhavish. I'm from Serum Institute of India, and very honored to be here among all of you, distinguished scientists and distinguished guests. Thank you so much.
It's a very nice question. So control human infection. In December 2023, we received the guidelines from ICMR. So after 4 to 5 years of continuous work with ICMR, so now we are open, but we also understand this control human infection will be the first in this country. So we are going step by step. The first one to build the infrastructure. So we are coming with a medical research facility. So where we are building the isolation units, which can accommodate up to 14 to 16 healthy volunteers. And those are in progress. The second step is to bring together stakeholders because there are also a lot of questions come up. So within first year of this, we will enhance the capacity building training and engage the stakeholders from the regulatory clinician and the industry partner together to open it for further talk. We are all, and the first infection model that we will work on is the influenza model because we have a vaccine, we have a drug, and the industry also has the capability to produce the challenge virus. So these are the steps we are taking one by one. So the MRC facility, Dr. Karthikeyan can tell more about the facility when it will be coming up.
I'm going to talk again about the chimp. So now that the guidance on controlled human infection models has come up, one part is the science part. Of course, we have the capabilities. Another very important component of this controlled human infection models is community engagement and public engagement to understand and stakeholder engagement. So we are going to initiate, I think the first couple of years is all going to be about stakeholder engagement, understanding the perceptions, any challenges that we foresee, and then trying to, so there are a series of stakeholder and public engagement workshops that we are proposing to do in the next year or so.
Answer this. There was an attempt here to do human challenge studies on a malaria vaccine. The DBT created a committee. I chaired that. After 1.5 years, half year's effort, it didn't succeed. We couldn't pass through ethics committees in India. There is no harm in this. Everywhere it is being done. But the problem was that some of the scientific questions which were asked, like once I was attending a meeting, and when it was found that aortus monkey is not a good model for malaria, Dr. Nasenvi said that humans are the best animals, do it in humans. And the human challenge studies were done in malaria and many other infections. But the main issues come up like malaria sometimes get internalized. There's a new science which everyone knows. So you couldn't answer some of the questions with the activists very strong in India. So it didn't succeed.
Just to not to take too much of time. So the MRC has, we have envisaged that facility to come up there. We are already in preparation for that. Like Dr. Nitya said, there are several issues before we start. And like Dr. Ganguly says, there is a lot of resistance, but I think we are at a point where now there is an ICMR guidance. So India is gearing up to it, and we will tread very, very softly. So it is not a program that we will do it in a hurry. We're going to take our time. And like Sweetie said, we will do the lowest risk chimp first, and where we are prepared and that there are already international guidelines and the attenuated strains are already available. So that's how we'll proceed. So it is a long project. So we have everything set up, but we will move slowly.
I would like to ask a question. My name is Anand Govinduluri. I'm a founder and CEO of Govind Capital. It's a venture capital firm investing in biotech companies, and I'm based out of Singapore. I came here because Dr. Manish Diwan asked me to come. From Singapore, I've come. I have two questions. First question is, do we have a TRL mapping of the various projects and the funding requirement for each project, which are suitable for commercialization? That's my first question. Because I think for an outsider like me who's coming for the first time and is getting introduced to the various innovative research programs for commercialization, it is important that we give a summary of the various projects which with TRL levels, readiness, and those which what kind of budget requirements are required to fund those projects going forward, because the future is all about public-private partnership, as what I've seen in Singapore. Second question is, what is the motivation of the institute to promote academic spin-offs? Because I was last 2 weeks back, I was in Cambridge. University of Cambridge has produced, and in my interaction with the Vice Chancellor, they said that the University of Cambridge has produced 10 unicorns in 10 years. And I then very innocently asked the Vice Chancellor, he must be having a very big budget. He said, no, it's only 50 million pounds. So that speaks volumes about the ecosystem and also the culture for producing academic spin-offs. So I don't know whether there is a culture of academic spin-offs from THSTI. I would like some answers on these two questions. Thank you.
Thank you, sir. So I will go for the answering the first question. So before we initiated the discussion with academia industry partnership, so this is the first thing, and I think BIRAC has already visualized this thing, the technology readiness label. So when we go for the product development, so apart from the research we are doing, so at least in the vaccine part, we have different TRL labels. So 3 of the vaccine has reached to the TRL 6 and 7, and it is with the industry collaboration. And 2 of the vaccine has been funded by CEPI. So we know that for this vaccine to go for the phase 1, phase 2 clinical trial, what will be the cost? And it's a huge cost. We do acknowledge that. And we know that even though there are different, that's why with the different TRL labels, we need the industry, government, academia, making a golden triangle and the clinician to take it further. Thank you.
The question about spin-offs, it's a complex question. We are a government institution. But having said that, there are three startups which have emerged from THSTI in the last 14 years, although we've made several more technology transfers. The modalities are not very well defined. Secretary DBT is here. These things are evolving. So there is a culture of startups as well here, but the way in which we set them up is going to be quite different. And we are working all the time with government. So it can't be like Cambridge, but we have been successful. If anybody else wants to, Dr. Gaurav has one. So if he wants to add.
So Department of Biotechnology has a policy, and so THSTI has 3 startups, and there are provisions in the agreement what kind of support THSTI is providing. And we have also space in the campus, which is at very nominal cost, and access to infrastructure. So all the DBT institutes now have policy that any startup can go pay and use the high-end equipment. So all those resources are available to us.
Just to add one piece here. So Department of Biotechnology has a clear mandate that technology needs to exit from academia. And there's a large biotech ecosystem that has been created and facilitated through VIRE. At present, 8,500 biotech startups are in the country. Next doors within the campus, we have a bio incubator, which is supported by activity. And if you look around, all the researchers who are sitting here, they are the potential people to create those seed such startups. Within the faculty, I think, as Dr. Gaurav mentioned, 3 startups have already originated. And I think more provisions for faculty, advisory roles, etc., mentorship and founders. I think those are being clarified, which Dr. Karthik highlighted. We can deliberate more on this and probably will give some overview also in the NCR biotech cluster, how we are envisaging that it would evolve and grow. Thank you.
Thank you everyone for the engaging session. For the next presentation, please welcome Dr. Manish Diwan, Head Biofundry, NCR Biotech Cluster BioRack, on stage to share the information about the upcoming facilities at THSTI.
It's still good morning. Very glad to see such vibrant energy sitting, a positive energy in the hall. Thank you everybody for joining us. What we think is that there is a vision that within this arena, we can create India's first largest planned mega biocluster. I think the industry people who have come on weekend with a focused intention to learn and shake hands with the academic counterparts, and the kind of overview which we just heard from our colleagues through what is the technical strength of this platform and this institutional setup under Department of Biotechnology. So let me give you a little broader vision that is driven through Department of Biotechnology to promote bioinnovation and biomanufacturing. And this NCR cluster, how do we think it can lead in the future? So if you look at any bio cluster worldwide, there are 4 elements that are fundamental to this. One is the R&D anchor institution where the new science emerges and it progresses forward for translation into products and tangible outcome. That is governed through policy and incentives, which involves support system like government policies, departments, ministries, etc. The third is the patient innovative capital to support such an ecosystem. And that is where both public-private investments come into play, which promotes that. And fourth, which is very important, critical parameter is the trained human resource. And I think which has experience of not only discovery, but also as the for the development as well. So if you look at Mass Bio, Boston, you have in a very close footprint, walkable distance, you get to see a high-end research being done in institutions funded by governments such as NIH, $69 billion assets being managed in that small footprint, uh, both from private investment as well as government, plus a large presence of R&D institutions from the industry side. Major industry footprint is there. It doesn't happen overnight. So for example, Singapore's Biopolis, when it got initiated in early 2000, it took time, and each passing year added new number of companies getting seeded in there, as well as more number of scientists and human resource which got seeded in that cluster. So we think that in this NCR biocluster, can it evolve as a India's largest biocluster in the NCR region, where we are sitting. This also converges 15 autonomous institutions of Department of Biotechnology, which have been integrated under the BRIC. So you get to sample and get closer to the science which is being done, not only in one institution, but a ensemble of 15 institutions. The scholars which within this area, within this campus, currently there are 800 plus researchers who are already working, and we got a glimpse of what is being done in THSTI just before this presentation. Core strength, we already heard a few technical presentation. I think I like the Dr. Ramandeep's summary presentation, which encapsulated everything in very crisp form. So it gives you the fundamental core facility strength, and each individual PI shared what individual laboratory is doing and focused on. So I won't go through that. DBT also initiated another PhD program, which kind of integrates all 15 institutions. And that is the next generation of industry-ready trained manpower, which is in making. So first batch would come out in August. Because of the locational advantage, we are very closely sitting with allied expertise. Engineering from IIT New Delhi, which is closed door, IIIT, we are also surrounded with hospitals. So we have the clinical express, not only within next doors, but surrounding areas. There are business schools like FMS, ISP Mohali. We have investor platforms operating through the NCR region. All government regulators are also close by. The Faridabad, Gurgaon area offers industrial zone as well as residential zone. We have easy accessibility. This place is located on a national highway, about an hour's drive on good time from the airport. At least Sunday, I hope it was more smoother. As we just mentioned, Department of Biotechnology, BIREC, has facilitated startup ecosystem in the country. So you can see there's already a consolidation in the north side. So all those startups who are seeded and incubation centers, some of them are represented here. They are also looking for facilities where they can touch base with the basic translational science that can be advanced, and infrastructure where they can move from POC to pilot and further pre-product development. This is the panoramic view of the cluster. We are at THSTI over there. So this is 200-acre campus, out of which only 40-acre which has been populated as of now. So there is ample opportunity which is sitting unrealized as yet. And as we move along, we heard about a medical research center that we want to seed up. That's coming up right there. We spoke about ambitious controlled human infection challenge studies. We want to drive towards the phase 1 capacities, doing the safety and dose estimation studies, clinical pharmacology there, so that the academic leads as well as industry, they can work together and progress such advancements into clinics. We are also coming, there's also another unit which is translational research facility, which would provide next order facilities to move towards piloting and pre-product. There would be clean rooms, GMP, and some of these translational leads that are mature, they can be further progressed in technology research facility. The open space that you see in the hills of Aravali, we want to bring in and invite R&D industries to seed in there, startups and SMEs to have their independent units, plus utilizing common services. So 4 elements essentially. Whatever we heard today, we want to expand this capacity to produce R&D and translational leads. So we want to expand the R&D capacity of this cluster. Second, we would like industry to come more closer. You have come in person. We would like industries to actually set up their units, R&D setups and facilities here, and startups, SMEs. That all ensemble would be supported through common access infrastructure and other supporting stakeholders to seed into the system. I think we already heard about TRF and MRC. I won't spend more details on this. So what could be the models of engagement with the industry, THSTI or this cluster for the R&D activity? We would like some lighthouse companies to come create their global capacity centers here. Industry people, they can have joint programs, sponsored programs, exploratory projects that can be done here. They can also have access to the emerging leads either through startups or from the PI's labs. We would encourage industry to sponsor projects, PhD scholarships or postdoctoral positions. Why would industry do this? We believe that industry would have access to the R&D leads much earlier before they get published and released outside. They would also have access to trained manpower and an opportunity to gain competitive edge for the innovation. And while engaging with the academic partner, it also opens up to you the funding possibilities which can come from government, private, as well as multilateral or G2G alliances that we promote. The cluster would also gain in many ways once these alliances happen. For example, they would get a product development partner. They would be able to out-license their leads. There would be an opportunity to gain insights how the development happens so that valuable experience could be gained. And I think overall, we will be able to undertake much higher value projects when we work together. When it comes to setting up infrastructure, one can do in 2 formats, which is like build, manage, and operate in leased land. Other way is that we are creating these warm shells like TRF and MRC. Come join us and populate that. There are examples elsewhere in the globe which have worked very well. We are open for many public-private partnership models. These are just the broad view. So together, I think this NCR biotech cluster is in making. We would evolve it as Biosciences Innovation Park with the help of all of you. And we look forward to changing the landscape of this area in next short time. Thank you very much.
Thank you, sir, for sharing your valuable expertise with us. Please join me in welcoming our distinguished guest on the stage. I request Professor Karthikeyan to kindly accompany our esteemed guest for the welcome. Professor Vinod Paul, Professor Nirmal Ganguly, Dr. Rajesh Gokhale, Dr. Alka Sharma. Professor Karthikeyan, kindly welcome the guests with a small token of appreciation. Professor Vinod Paul. Professor Nirmal Ganguly. Dr. Rajesh Gokhale. Dr. Alka Sharma. Thank you, sir, for honoring our guests.
Okay, so as the focus of today's talk, today's session basically is innovative research with translational potential. It is my immense pleasure to invite Dr. Alka Sharma to share her thoughts on translational and innovative research driving science to solutions. Dr. Alka Sharma is currently Senior Advisor in Department of Biotechnology, Ministry of Science and Technology, Government of India. She is dealing with research and policy issues in the emerging areas of medical biotechnology. She has made significant contributions in the mid-tech innovation with national and international partners, early and late translational research on stem cells and regenerative engineering, setting up bioclusters across the country for innovation-driven research. Please welcome them.
Professor Vinod K. Paul, Member Niti Aayog, Dr. N.K. Ganguly, former DG ICMR, Dr. Rajesh S. Gokhale, Secretary DBT, DG BRIC, and also Chairman of BIREC, Dr. Manish Diwan, who you all have heard him, all the dignitaries of the dais, THSTI family, industry experts, and all the participants. Good afternoon to one and all present here. So it is indeed a pleasure to be part of the first industry meet at THSTI today. And I am going to just provide you a very brief overview on translational and innovative research, which is a key driver of science to provide solutions. So here I would like to congratulate the THSTI family for its 15th Foundation Day. Tomorrow they are going to celebrate. And it's so congratulations to all of you on the eve of Foundation Day. And it's such a nice to see this kind of positive energy in this hall on the eve of 15th Foundation Day of THSTI. So for those who are not new to this, maybe the ecosystem and also coming first time to THSTI, the interim, it was started in the interim facility by Department of Biotechnology in 2009. And here you can see that foundation was laid by the then Secretary, Dr. M.K. Bhan, in July 2013, and that was 15 July. So as you all heard since morning, the kind of translational research and clinical research is being conducted at THSTI, and also the facilities by Dr. Ramandeep. And you can just see that these are the facilities available here in THSTI for the THSTI faculty and also for other scientists and clinicians and industry who all are doing research in these areas. So I'm not going to repeat this, but you can just see right from the vaccine development, biorepository, and also you heard CDSA from Nitya and the kind of the role CDSA is playing in promoting and supporting clinical research at THSTI. Morning also, the director, Dr. Karthikeyan, he in his overview, he provided the gist of that kind of the activities are being conducted at this place. On your right side, you can just see the focus of this institution is on promoting, nurturing the basic and clinical research in an integrated manner, preclinical research, and also cohort studies. And we also heard precision biotherapeutics from few scientists who presented, Dr. Bhaptos and others. And Dr. Jayanta mentioned about monoclonal antibodies, which is one of the important component of precision biotherapeutics of the biomanufacturing initiative, which Department of Biotechnology has taken so far. And then biotech also the innovation park that is under consideration. So I just, since this is an industry meet, I thought that we should just discuss together that where do we stand today and what is the way forward and what are the opportunities available for translation innovative research in our country. So many of you are maybe aware that global biotech industry has grown over the past few years more than 5%, and India contributes more about 3% of its market share to the global biotech industry. And India is ranked 12th in world in biotech sector and third in Asia Pacific. We are the largest vaccine manufacturer. We have emerged as the third largest startup ecosystem globally. And we have created a strong biotech research ecosystem across the country. And Department of Biotechnology is the first agency to set up BIREC as its industry academia interface agency to promote and support public-private partnership. On your right side, you can see the number of companies, biotech startups, products which have reached to the market, and biotech, the bio incubators, teaching and research infrastructure and institutions which have been created in biotech sector in the country. And currently we have crossed 137 billion bioeconomy, and that data was up to December 2022. The data for 2023 is under compilation. So, and the target is to achieve 300 bioeconomy by 2030. So here I would like to mention that the government is playing an important role for laying down strategies, formulating policies, creating ecosystem to facilitate end-to-end processes, and also creating critical mass of trained skilled manpower, and also addressing regulatory issues from time to time. So biopharma accounts for the largest share of bioeconomy. And you can see here there's a huge jump in the rank of our country from 81 in 2015 to 40 in 2022. And this is now being from 81 to 40, and it is being maintained in 2023 also. And as mentioned by Dr. Manish, that India as a global innovation hub now has resulted into creation of more than 8,500 biotech startups up to 2023. So Indian bioeconomy has registered 29% growth in 2022, and you can see the growth of bioeconomy from 2020, that is from 86 billion to more than 137 billion in 2022. So bioindustrial segment was the largest contributor to the total value of bioeconomy, followed by biopharma and bioagri, and then bio-IT and research. So as you all know, maybe that you have heard from other previous speakers also, the Department of Biotechnology has established Biotechnology Industry Research Assistant Council, that is BIREC, as its academia and industry interface agency to promote and support public-private partnership. In more than a decade, the BIREC has now created a vibrant biotech innovation ecosystem across the country through its various schemes and programs, and established bio incubators across the country. You can see the numbers here, established national international partnerships, provided support to biotech startup entrepreneurs, and also more than 800 biotech products have reached to the market with the support from BIREC. So I'm coming to this that department, many of you were the part of the brainstorming session, which was conducted by DBT at various places in the country. So what is the opportunity now for all of us to drive in the country in terms of translation, innovative research leading towards the product development? So fostering high performance biomanufacturing has been initiated by Department of Biotechnology under the leadership of Dr. Rajesh S. Gokhale, Secretary DBT. And I just want to share with you that this is the need of the hour for the country, because the climate change, unsustainable pattern of material consumption, excessive resource utilization, and waste generation, we all know that they are global threats and require concerted sustainable interventions. So biomanufacturing has the potential to address these challenges by leveraging living systems to create new sustainable and efficient manufacturing processes with reduced environmental impact. And also this will help in advancing medical treatments, promoting bio-based products, and supporting innovation in various industries. And these all are possible through various biotechnological approaches. And then I'm going to just share with you what all are the thematic areas which we are focusing under the umbrella of biomanufacturing and biofoundry. So this initiative is aligned with our national initiative of Government of India. This includes the emphasis on green growth which was announced in Union Budget 2023-24, and also it is aligned with the Honorable Prime Minister's vision to make India a net zero carbon economy. And the Honorable Prime Minister also announced that Life for the Environment in October 2022. And this year also, there was a biomanufacturing and biofoundry has been announced as one of the schemes under the Union Budget, that was 24-25, that was announced in February. So the message is very clear that this will foster a future that is both sustainable, innovative, and responsive to global challenges. And this has potential to create new jobs and intensify entrepreneurial momentum. And this is the bio vision for Bharat during Amritkal, that is India at 2047. So just to give you the genesis, that the national consultation was organized by DBT to discuss this area in detail, and also to identify the thematic areas that which all thematic areas can be brought together under this umbrella of fostering high performance biomanufacturing. And this was attended by all stakeholders that include industry, academic institutions, and startups. And these are the 6 thematic areas which have been identified based on a discussion held during national consultation, and also we had interministerial consultation also, and the 21 ministries and departments were consulted before finalizing these thematic areas. So basically, first one is bio-based chemicals and enzymes. This also includes biopolymers and APIs. Then the second one is functional foods and smart proteins. The third one, precision biotherapeutics. You have heard from the faculty from THSTI since morning. So monoclonal antibody is being driven by THSTI under precision biotherapeutics. Apart from that, there is a focus on cell and gene therapy. That was also a talk from Dr. Amit. and mRNA therapeutics. Then the fourth is climate resilient agriculture, and followed by this carbon capture and utilization, and futuristic marine and space research has been added as a new component in this, and that what kind of biotechnological approaches can be applied to these sectors. And these thematic areas will be augmented by bioenablers, that in the form of bio-artificial intelligence, bio-AI hubs, and also biomanufacturing hubs in public-private partnership and international collaboration. Just would like to share here that DBT has established collaboration with National Science Foundation, USA in this, and then signed the implementation arrangement for this sector. So we all know that biomanufacturing hub, this is very, very important to drive this biomanufacturing initiative. And this will facilitate translation from lab to market for scale up, and that up to pilot, and then followed by pre-commercial manufacturing scale. So the presentation made by Dr. Manish Diwan, so most of the actually that bringing stakeholders together for this common goal, it is going to add value, and this is the right place where we can start this kind of integration of the facilities. So here you can just, as I mentioned that after having all the identification of all 6 thematic areas, it was discussed that it would be better to have brainstorming session on each thematic area with all stakeholders, including startups, industries, clinicians, academicians. So what kind of roadmap we should have, which there is some gaps and challenges. To identify gaps and challenges, these brainstorming sessions were organized across the country. So total 17 meetings were conducted, and now actually we are very clear that which is the path we have to take in this particular area. So here, in conclusion, I would like to say that these efforts are going to certainly going to bring technology revolution with new wave of innovation in engineering, biological systems, and the collaboration between industry and academia will foster innovation, knowledge exchange, and technology development. And this will help in positioning India as a global biomanufacturing hub for biotransformation. There will be paradigm shift towards a bio-based circular and decarbonized economy, and this is certainly going to foster surge in employment and intensify entrepreneurial momentum. Ultimately, it is going to accelerate to achieve India's 2030 goals of achieving 300 billion bioeconomy and national economic goal for 2047. Thank you.
Thank you, ma'am, for your engaging and insightful address. I request Professor Karthikeyan to kindly felicitate Dr. Alka Sharma. Ma'am, we need you on the stage. Thank you, sir. With a distinguished career spanning several decades, Dr. Rajesh Gokhale, Director General BRIC and Secretary DBT, has played a pivotal role in shaping policies and fostering collaborations that promote innovation in biotechnology. An alumnus of IISc and Stanford University, Dr. Gokhale has contributed significantly to advancing research and development initiatives in the country. Recipient of several prestigious awards, to name a few: Shanti Swarup Bhatnagar Award 2006, Infosys Prize in Life Sciences 2013, his expertise and leadership continue to drive the growth of biotech sector in India. Please join me in welcoming Dr. Rajesh Gokhale to kindly share his perspectives with us. Thank you, sir.
Namaskar and good morning to all of you. Lovely to be here today amongst all of you. It's really exciting to see all of these people from all across the country, and somebody even mentioned from Singapore as well. Actually, we're very happy to be having you here, actually. I didn't know we have an international gathering today to celebrate this fantastic success of this THSTI RCB campus, what Department of Biotechnology initiated 15 years back. Professor Vinod Paul, member Niti Aayog, Professor Nirmal Ganguly, former DG ICMR, Professor G. Karthikeyan, Executive Director, THSCI, my colleague from DBT, Dr. Alka Sharma, distinguished guests in industry and academia, esteemed colleagues from DBT, BIRAC, BRIC, THSTI, RCB, directors of our DBT institutions, and ladies and gentlemen. I'm really delighted to be here today amongst this gathering where we see best minds of academia and industry coming together to galvanize momentum of the shared vision to do Amrit Kal, drive India towards the futuristic goals of the activities. If we really want to see what are the two major driving factors that are going to drive our country forward, and those are, I can really in two precise words, I can say one is a digital growth and the second one is a green growth actually. Primarily because every industry will have to work in a way such that it changes its style of functioning from what we have today than what we would have in future in next 20, 25 years. The technological speed with which things will change are going to be neck deep and very breathtaking actually, that we ourselves sometimes find difficult to adjust to actually. And I think that's where what really India's ecosystem provides a huge opportunity for us to do. If you really look at in the last 2 decades, what India has done fantastically well now is have invested in capital, infrastructure like this, have invested in people, and then invested in knowledge base that is now almost comparable in par with several international standards. What we're really looking at is an opportunity to break the ice and convert all these activities into a driver of economic and societal development for the country's future. Actually. Now that's easy enough if you really think so, but it's an amazing task that requires enormous amount of people to come together. And in fact, sync ourselves, or we call it because of the name of this, cinch ourselves actually into a kind of a time where we can really carry out these activities from the futuristic goal perspective. I think the landscape, particularly from a healthcare ecosystem that we are talking about today at THSTI's perspective, I must say that there are amazing things happening in the labs, not only in DBT institutions, but in CSIR, in DST, in ICMR institutions, which we all need to learn to capitalize actually. What it requires is a little bit of mentorship. It requires some partnership and lots of investment. And the investment doesn't come until and unless all of us bring together all our thoughts and ideas in a way that creates huge pool of money, which can really bring us into an era of what we talk about rapid innovation ecosystem. I think that the technological challenges are very many, but opportunities are even more than that. And that's what makes it such an exciting journey to be able to do these activities. As you've already heard from 2 of my colleagues, I'm not going to reiterate and I'll be just short in my own thought processes. I think Dr. Alka Sharma from DBT and Dr. Manish Tiwan from BIRAC has given you the larger vision of what DBT is looking at as an opportunity cost of activity. I don't think so ever in our journey of our country's independence that we had this opportunity really to be able to come and change the country's progress. Many events we attend, many events we really come together and think of it as, activities. Believe me, today's event is a transformative step in the future of our country's progress. And that's where I think we should all really feel important and also provide as much as possibilities from our own perspective to be able to make this happen into a real world challenges that we are talking about. Networking is the key mantra to success, actually, and it still continues to be despite all the media platforms and everything. Share your ideas, listen to each other's, explore potential synergies. And what we've been able to do through DBT is we had several institutions, about 14 of them all across the country, in all top institutions, believe me, that at the high end of technological innovations, actually. And but they are all scattered, and there was no clear perspective in terms of how to bring them together. And government pushed us to really put them together into what we call Biotechnology Research Innovation Council, actually. And this we have done this, bringing all our capacities and capabilities into one single platform. What it does, it makes the biggest body in the country in terms of its bioscience scientific growth, with more than 1,200 PhD students doing fantastic work all across the country, actually. And I think that makes us very, very strong, very powerful, and very, very impactful in terms of deciding and taking bold decisions of what we want to do. What we really have to do is to leverage its capabilities that are present in all systems and work together towards what we would like to see in future systems to come. Several programs, including novel program of what we developed recently for PhD program as well, actually, what we call I3C. Actually, it's called Ideate, Innovate, Immerse through Collaboration. And the whole perspective is that the single PhDs these days neither bring value to students, nor to individuals, nor to the society. And therefore, the challenge for us is how the student really does this cross-cutting understanding of various ecosystems at very early on, such that they are ready to work on a collaborative mode in a larger programs. And under these programs, what we have done is we've kind of put together our thoughts and starting to address national problems through PhD students, such that that becomes empowered at very early part of their careers and activities. I think BRIC THSTI, a couple of words about since we are here, has always played a pivotal role to translate groundbreaking research from practical health solutions in various aspects. You've already heard about all of them. You can see almost every single vaccine in this country has come through some or the other intervention that has come during the COVID times is the THSTI's intervention. I think we should all be proud about this, actually, right? At the time when the world was suffering, the government institutions was willing to part and work together with private industry. And as you can see, of course, private industry gets all the good backing. But that's important to feel part of it, actually. And I'm very, very proud of DBT institutions have put together. Similarly, the INSACOG activities that has been going on all across the country of looking at monitoring the viral, what kind of strain it is traveling around the country, how is it doing it. It's for the first time coming together with all the ministries. And believe me, it's not just DBT, but several ministries came together. And we put together a team to actually sequence more than 3.5 lakh genome sequences of various viral strains that were collected all across the country. It's no joke, believe me. It's a vast country. Lots of things needed to be done. But we now have a data that is available in the data center that is present in this campus itself, called Indian Biological Data Center. So you can see that this campus now actually has everything. Yes, it has an academic ecosystem with the Regional Center of Biotechnology, THSTI's Translational Health Sciences Facility, IBDCs, animal models, biosafety facilities. What we really want is industry to have a vibrant ecosystem for them to succeed and for country to succeed further, actually. I think we can only together push the boundaries of what is possible and what we can do together for the science and technology innovation ecosystem to build a healthier future. I would just request all of you to embrace this opportunity, make the best out of what we have today, and I think feel proud of the future of this country. Thank you very much. Namaskar.
Thank you, sir, for motivating us and sharing your visionary thoughts. I request Dr. Karthikeyan to kindly felicitate Dr. Rajesh Gokhale. Now I request today's guest of honor, Professor Nirmal Kumar Ganguly, to kindly address the gathering. Professor Ganguly is a microbiologist and his major areas of research include tropical diseases, cardiovascular diseases, diarrheal diseases, encompassing disciplines of immunology, biotechnology, and public health. He was formerly a distinguished biotechnology research professor, Department of Biotechnology, Government of India, and has awarded over 100 awards, including 7 international awards. He has been honored with prestigious Padma Bhushan Award in 2008 in the field of medicine. Sir, it is an honor to have you here.
Thank you, Dr. Karthikeyan and Amit, to invite me in this August gathering. Professor Vinod Paul, Professor Rajesh Gokhale, Dr. Alka Sharma, Dr. Jayant Bhattacharya, Dr. Bhavtosh, Ramandeep, and all the distinguished gathering who have gathered here. I've never seen this hall so full ever in my life. So let's stop it very interesting. So I've never been introduced properly. Actually, I was a chemist who became a microbiologist, but then did fellowship and worked in various clinical departments and became an immunologist in Japan, and then did my degree in public health to make myself complete. So I don't know what I am today. So that actually, if you have some basic science and in life, in the pure science, you do better in your medical life. So I will tell you about new experimentation. Today, when we are talking of translation, there are 2 people who are here who have succeeded. I have never seen, it was a revelation for me to see the current THSTI. It will go very far, I'm very sure, because the overall structure is very nice. Actually, myself and Dr. Bhan, I will say Dr. Bhan and myself, I had hours of walking and discussing about the THSTI in the Asia Village complex. But I think he also perhaps never imagined, and I never imagined that this will take this shape. So congratulations, Dr. Karthikeyan. And we have something common with Dr. Karthikeyan. I started my life with rheumatic heart disease, and this perhaps he will start something over here, and I will be very happy if he does that. The other person which is here, and I think he is an ultimate translation person. That is the transformation of IGI with Dr. Brahmachari and Dr. Rajesh Gokhale, which happened. That time is not there to really see what I saw there, but it was very, very transformative. So the enabling mechanism of BIRAC, TDP, inimitally, and many other enabling mechanisms which the government of India set up have really helped to foster this and nurture this. Dr. Alka really talked about BIRAC success very lucidly. I was born in 2007 November, and so there's not a joke. Actually, I started my new life in 2007 November, and still continuing it, and will emphasize the problem. And if you don't believe, you go to the computer and see that I'm publishing even today. Perhaps after Dr. C.N.R. Rao, my publications at my age will be quite impressive. So this is one of the age group which are lost in India. They are mature people. They have translated things. Many of them perhaps will go to fruition. Whatever mechanism we have to give them a second life doesn't exist. So I started an experimentation. I was the RC chair of almost all major institutes in India. or was a scientific advisory member in some of the other institutes, including health institutes. So I have seen there is something lacking there, that we have excellent institutes which are manned by PhDs. We have institutes apart from All India Institute, Vellore, etc. We have health institutes which have a capability but are not able to deliver. This meeting didn't happen. So I started a new experimentation. I wanted to develop research departments in the corporate sector. And at the moment, I employ 40 scientists whom I pick up, and have 4 research facilities. One of them is 4 floors in a hospital, which is unheard of. And when we were looking at infrastructure, which Amanti was showing, we have everything. We have multi-omics, high-resolution mass specs, NGS, BSL-3 facilities, production, 2 or 3 production, CGMP production facility, everything, imaging, confocal, you just name it, and we have it in these corporate hospitals. And the model was very, very different. They don't invest unless there is a return. So the model was that whether we can give them a return to their investment. And this will be a pleasure to really show that what happened there. So I will be spending 2-3, I have 10 minutes, 2-3 minutes to develop a philosophy, but a few listing which was successful and came out of this new incarnation. I have put up 4 vaccines in the market. The world's first subunit rabies vaccine, the quadrivalent influenza vaccine, immunoadjuvant vaccine for Mycelex, which is for lung cancer, and an adult septicemia immunoadjuvant. All of them DCGI approved. Other ones which are in the DCGI table are the quadrivalent HPV vaccine, the shingles vaccine, the chickenpox vaccine, and in the drawing board is now injectable new generation rotavirus vaccine. So these are all realities. They are all in the market or in the DCGI table, and this has happened with these interactions. The other areas, like I will just touch it quickly, that because THSTI project presentation was very good, I will just list some of them. Like if we come to Bhavtos's presentation, so we have a very strong microbiome facility. So we have 2 different areas which we have developed. One was that some of the lung cancer patients didn't respond to checkpoint inhibitors. But when you looked at the microbiome, and if you did FMT there, the fecal microbial transplant, then some of them from the stable disease started responding. The same way for the colon cancer microbiome, the ones who didn't have the good microbes, if you change them, they started behaving better. And as Bhavto said, I had one of his students, and she had come up with a real ideal donor. And a combination of these bugs, which if they are assembled, they will be useful. And diagnostic kit for dysbiosis. So the microbiome area in these private sectors are developing very strongly, because the model is that puts PhD students in the clinical settings and pick them up. So all of them were picked up, all of them were salaries which they deserve, and make them all happy, independent. They work under no one. They have an ownership of whatever they do, and they produce. So this, in the same fashion, if we move further, like in the diagnostics arena, we have one added out of the 16 autoimmune antigens, we have added one antigen. We have added a test which has gone to the company now, which detects glioma, high-grade glioma, and this is a blood test, and it goes with the grading of glioma, and if the glioma has removed, it changes. So there are several such diagnostic systems, particularly in the biomarker development arena. We have invested very highly, and this has been just published few days back. For the pulmonary hypertension, we have developed 5 markers which could be used for surveillance in the COPD who are going towards pulmonary hypertension. If we really, I will take one more example, like in the arena of monoclonals. At this given moment, one of the major need is that whether monoclonal will work or it will not work. So we are creating matrices like in the lung cancer. We now have a series where 50% have survived for more than 5 years and more by identifying who will be responding. So the biomarker discovery in the various arenas, whether they're autoimmune disease, whether they're cancer, is really very good. To commercialize them will be one of the major challenge, but that is what we need to achieve. Finally, I come to one more thing we are doing, like CDSA might be interested. We are running with Dipsaru 2 clinical research courses, and every one of them get employed in a very nice setting. They get a hospital exposure along with the clinical courses. The basic mechanism that we have a PhD program, robust PhD, and we have an internal funding mechanism. Finally, I come to a philosophy in the cell therapies. Few of these which I listed, I had collaborated with the THSTI, with Amit, with Bhavtosh and others. And this is another area where we will like to collaborate. Like in the cell therapies, I have found out 2 major problems. I was in the front seat because I was in the governing board of the Tata Memorial Center for 18 years. And when this CAR-T cell therapy was developed, it was very good and robust. The first time it developed with Bombay IIT and the Tata Memorial. Again, very nice combination. However, the price is 30 lakhs if Rahul Purwar gives it on a concession. Otherwise, it is 45 lakhs. And when you put the people in the hospital, in the ICU setting, 1 lakh is that every year it ranges. And there are a lot of side reactions, they have to be treated free. And if somebody dies, you have to prove that he dies of the natural progression of disease, because only 30 to 40% survive. And out of all the cases, only 30% qualify to be treated. So there's a huge ethics problem. Tata Memorial is able to do it because they have huge donations, and these donations, they subsidize the patients. But how do you choose a patient? Like in the transplantation arena, there is a guideline and there is a waiting list. You get an organ if you qualify for that guideline. Here, how you do that? There is a person who qualifies, but there is no way he can afford to do this. He cannot afford 45 to 60 lakhs rupees for the treatment. So what Amit is doing and what we want to do with them, we have set up a full floor for the CAR-T clinical setting. We have started getting something and getting experience of what happens after you treat. Like we have created a toxicology panel. We are creating a panel where we could do whether it is surviving or not, whether the CD2, CD15 cells survive or not. We are collaborating so that with the companies, so that the CD2 and CD15, 2 cytokines regular are produced at a cheaper rate, and also create a series of lentivirus and other vector systems which are very, very costly, I was very happy to see that Amit has created a series, and few of the series we are trying to create. And ultimately, this marriage will be very, very good, because in a private setting, what happens in one of the parts is a foundation. What happens, that decision making is very quick. You decide and you do it. You want to hire a person, you pick up, you pay the person, and the person comes. Only thing that you need to make the person happy, provide an environment, and few of the experiments now are happening, but they have not gone into medical center. Like Dr. Agarwal went to Ashoka, Sasidhara went to HD Goenka perhaps. So, but those settings are not the ideal settings. But in this setting, if we could really collaborate, I talked to Amit and Dr. Karthikeyan, and we had a discussion. If we could do it, we could create a system where we could develop the new system. At this given moment, we are having two Miltenyi expanders. But what we are doing with Milten is for the myeloma treatment, because the ALL and other treatment at this given moment, they have already given, and they cannot give it to us. But with Amit and with such collaborators, we want to do this. So ultimate aim is to provide this treatment in 12 to 14 lakhs. And if it can be done, then you could do ethically, you could take from Prime Minister's fund and other funds and could treat everyone who deserves this. Thank you very much.
Thank you, sir, for your incredibly valuable insights. I hope it will guide us in the right direction. I request Professor Karthikeyan to kindly felicitate Professor Nirmal Ganguly. Thank you, sir. We have today with us Professor Vinod Kumar Paul, who is a prominent figure in India's healthcare and policy landscape. Currently serving as a member of National Institution for Transforming India, Niti Aayog, he has played a pivotal role in formulating the Ayushman Bharat PMJ, the Ayushman Arogya Mandir scheme, and Poshan Abhiyan. With a distinguished background in pediatrics and public health, he has made significant contribution to child health and development initiatives. He is well recognized globally for his leadership in shaping national health policies and strategies aimed at improving maternal and child health outcome. Dr. Paul had also played a pivotal role in the Union government's task force for COVID-19 pandemic response. His insights and vision continue to drive impactful changes in healthcare delivery and policy formulation in India. It is with immense pleasure that I invite Professor Vinod Paul to kindly deliver the presidential address to the gathering.
Good afternoon. Namaste. Respected Dr. Rajesh Gokhale, sir. Respected Dr. Ganguly, one of my mentors in my journey. Dr. G. Karthikeyan ji, Madam Dr. Alka Sharma, deans, faculty, scientists, and scholars, and indeed the entire family of THSTI and Biotech Science Cluster, NCR, Faridabad, and above all, esteemed representatives of the biotech and pharma industry of our country, and friends. I'm very, very honored to be here this morning and this afternoon. It has been a very heartwarming meeting for many, many reasons, several reasons. First and foremost, we are witnessing the great achievements of THSTI
THSTI has completed 14 years, completing today 14 years of its existence. It's now entering late adolescence phase. I'm a pediatrician, so now it will become late adolescence, and really, it is a time when we're moving with a certain trajectory, but the exponential phase is really all set to begin. And very appreciatively, you would have all noticed that we are now seeing clear products emerging out of this great institute, science emerging out of this institution, human resources, and truly it's playing a role, and it's increasingly taking a trajectory of playing even a greater role for human health in the country and the world. So first, a big applause for the birthday of THSTI. Thank you for your presence, particularly the industry, at this celebration. I also like to pay my humble tribute to my mentor, late Dr. M.K. Bhan, in whose name we have this auditorium, and it brings back many, many memories of the love and affection and the mentorship I received from him as a teacher, friend, philosopher, guide. And also, today we also celebrate the vision that he had set for this institution, and we are proud and we can report to him, I'm sure he's around, that we are doing well with the cluster here. This was his most favorite cluster. He made other biotech clusters, but then his real, I would say, most favorite cluster was the Faridabad cluster, as we used to call it at that time. And today he'll be a very happy person. So my tribute to him. I like to essentially make 3 points, but let me begin with the context which has been set also by my very distinguished earlier speakers. Alkaji spoke about it and Dr. Gokhale spoke about it. I think, we are passing through a very, very special period in our nation. We know it. We have dealt with a pandemic which has taught us many, many lessons for our security, for our well-being, for the challenges that could come. And with climate change, they will perhaps come more often and in a more severe shape and form. So one aspect is that we have learned that our health products, our health system, and our capabilities in solutions and countermeasures, therapies, diagnostics, vaccines, preventive strategies, and so on, have to be very strong, not only for us, but also the world looks upon us to deliver it for them. I just want to mention here that if we had not made our own vaccines on our own soil, just close your eyes and look at the scenario. We delivered 222 billion doses. And I'm privy to the fact that we actually had a total offer of 13 crore doses only from the world. 13 crore और 220 करोड़, ये कहाँ जाते हैं? सोचिए, अगर ये ताकत नहीं होती तो ये ताकत हमें बनानी ही है. You can imagine what it would have meant. The first year of vaccination program, a Lancet paper tells us, saved 34 lakh lives in India. It's a global other estimates, but India estimate was 34 lakhs and more. And imagine what it would have meant to the nation. Nobody would have come to our rescue. तो ये लेके चलो कि आत्मनिर्भर बनना है, लेके चलो कि जो यहाँ तक की सक्सेस डॉक्टर अलका ने दिखाई, इतना बेस है कि हम उसको अब ऊपर तक ले जा सकते हैं. डॉक्टर गोखले ने कहा कि ऐसा मोमेंट कभी कभी आता है, और आज यहाँ ये बात कहनी चाहिए. कोई नहीं आता. आप इमेजिन करिए 13 करोड़, या उसको सिंगल डोज भी थे उसमें से थोड़े से. सो वी कुड हैव ओनली वैक्सीनेटेड 19 crore people. And 19 crore is 200 kilometer radius from NCR from this.
So please, let's join in the journey of being Atmanirbhar. Second, the context of Viksit Bharat. We have set our eyes and we have the potential, we have the energy, we have the DNA, and we have the wherewithal to reach the level of a developed nation by 2047. The Honorable Prime Minister has given this call and it has caught the imagination of all of us, each one of us. We have to do our part. And in order to be there, our institutions have to be strong, our products have to be strong and perfect, and to be in plenty. And we have to be able to contribute our might to the GDP that would be expected at that time, the growth, the economic, let's say, envelope that we would require. It is our estimate that at that time, our economy would be the size of 30 trillion. For us to be there, it means we should be 30 trillion economy. It would mean at that time, our per capita income will be to the tune of $18,000 in today's value. Today, we are about 2,300 or 2,500. So imagine the journey that we have to take. And we have to contribute from our sector to that as well. 300 billion, as Dr. Alka said, is the goal by 2030. But we have to go well beyond that because this is where new technologies, new products, new global needs would emerge and we need to contribute. And therefore, this meeting is so very special. It's special for us to be Atmanirbhar, for us to shine and give our might and contribution toward Viksit Bharat. That's number two. And third, to be part and parcel of the global value system, the global supply system. Indeed, this would require scientists, this would require great PhD programs and so on. And then, of course, our work starts moving out from that vision. So it's a special moment. It's a special moment. 15th birthday of the HSTI, and we are going to make our plans, our vision to do so. And thank you so much, industry, that you are there, because as you've seen repeatedly, the pathway of an idea to a molecule to a product is incomplete without your help. So that's my now first point and first request. Please develop and make your products with the HSTI here. Please join us. You are making in India. Yes, we are now requesting specifically on behalf of the DBT and the government leadership कि आप DBT के साथ THSTI के साथ इस जगह पे हम सबके साथ मिलके प्रोडक्ट बनाइए और हम जानना चाहेंगे कि आपको इसके लिए क्या मदद चाहिए. And this, in fact, we have proven the point in our difficult journey of COVID-19. Didn't we work together? Didn't we help each other? So many essays were done here. Grants were made directly to the industry. At least 550 crore were handed over to the industry to develop the products, the vaccines to be specific. DNA vaccine was supported by DBT. mRNA vaccine was supported by DBT. The nasal vaccine was supported, and so on. So practically all the vaccines, were one way or the other, were supported with cash, with assays, and with partnership. And we also work together to ease out the regulatory pathways on a daily basis. We're talking to each other on a daily basis. So we have a model. We already have a model of collaboration. Academia, government, industry, we were all working together. So please, let's build on that model. Let that not only be reserved when a calamity comes, but let us do that on an ongoing basis. And we assure you that that's the philosophy with which the government is working. And we would really welcome you to continue to please take those steps. And you have some wonderful products on the scene. ये निपा antibodies बना सकते हैं क्या हम इकट्ठे? स्पीड के बारे में मैं बाद में आता हूँ. That's my second point. मलेरिया का डायग्नोस्टिक बनाए, कब क्यों नहीं बन सकते? और भी हमने सुना लैक्टोबैसिलस का प्रोडक्ट सुना, कुछ मेरे को समझ नहीं आया. I'm just a
पीडियाट्रिशियन। तो हमें ये बनाने हैं, हम आपसे यही कहना चाहेंगे कि आप जुड़िए और आप हाई वैल्यू हमेशा देते आए हैं। आप भी जानते हैं कि हाई वैल्यू से पूरा जर्नी पूरा नहीं होगा, विकसित भारत की तो जर्नी बिल्कुल ही पूरी नहीं हो सकती। आपको हाई वॉल्यूम के अलावा हाई वैल्यू को बनाना भी है, डेवलप भी करना। दैट ऑल्सो वी नो दैट वी कैनॉट compete with the Chinas of the world unless we do research and development. It is time for you all, for the private sector and the industry, to contribute more, both in terms of effort and in terms of resources for research and development for pharma and medtech. I may just mention here that there is one particular scheme that helps us also, but just for you to know that we have a scheme called PRIP, which is promotion of research in pharma and medtech, research and innovation in pharma and medtech industry. It has 4 components. 3 of them are for the industry and startups and MSMEs. I'd be happy to share more details with you, but here that money is available. And the first component in particularly is for academia and industry research, particularly for the national science laboratories. So please harness this. Tell us how we can help you. Reach out to me directly. We'll connect the dots for you. And that's the spirit with which we would like to work. The second point that I wish to make is the speed. Not doing things with speed is an opportunity cost. Not doing things with speed would mean deaths, ill health, loss to the nation. Of course, everything cannot happen instantaneously, but the fact remains that we must learn to work with speed. And that brings me to the notion of a 100-day mission that we have learned, we have now recognized, and the world has recognized, is the path to showcase how priority products have to be delivered, of course, in the wake of outbreaks and pandemics, done. But that's an example of how speed is important. And speed is important for CAR T cells, sure. Speed is important for, anti-TB drugs. But speed is also particularly important for those areas of our work where we should be ready for future pandemics. So I'm bringing in a specific focus for those products, those countermeasures which are related to potentially future pandemic, future outbreaks. And here, the list that you saw, I'm happy to note that several of those products, I saw them being listed as being in various stages of development in this very institute. So we have worked on Nipah, for example. We have worked on some of the other viruses. You saw number of virus families and SARS. I'm sorry, I'm told MERS we have, and others. So the notion of 100-day mission is the following for the students more than anybody else, is that during COVID, the world delivered vaccines in about 9 months, 350 days. India did it in the same period. We did it just with the world, not after them. But as we heard from Madam, that there were 3 decades of work prior to that, and we could escalate, right? There was a preparation of sort that was there, and then we were lucky. The thing started to work, and so many vaccines were put on the table, and India ultimately produced 8 vaccines, licensed 8 vaccines in all the 5 platforms. No other country has done it. So it happened. Now the world recognizes that in such an eventuality, we will have to deliver vaccine, and we should deliver vaccine in 100 days. 100 days. In first 100 days of COVID pandemic, in the world, 25 lakh cases have happened and 2 lakh deaths had already happened. 100 days, already happened. Okay. Now, this is still so late as that. And then, of course, many more happened before the vaccine. The point is, first 100 days are critical. And now we will be competing out with the people who are already preparing to deliver this in their own countries. So my point is we have to be ready with our own 100-day mission. Having learned that nobody else will come to our rescue, our own 100-day mission for future pandemic has to be one important strand that we all have to work. So the products that would prepare us to face future pandemics, disease X, have to be readied now to a stage. So that's one part of the work. I'm not saying this is the only way. There are a lot of other things to be done. There are other diseases to be tackled, the chronic diseases and so on. But I'm trying to say the test case for our collective work is also an area where for future pandemic preparedness, can we work together to pick on certain products, certain countermeasures, and I heard a few of them already today, on which can we deliver them in 100 days? Can we now accelerate Nipah monoclonal antibodies to be delivered in 100 days? If we can't do it now, will we do it in the wartime? The point I'm making is, with that kind of a synergy, effort, energy, Josh, can we demonstrate that we have the capability for things that are already familiar to us? I want to put that challenge to you. And therefore, specifically, let me offer to say, for Nipah and monoclonal antibody, 100 दिन का प्रोग्राम बनाएं क्या? मलेरिया का एंटी वो जो टेस्ट आपने कहा कि इतना अच्छा है, and malaria we have to eliminate by 2030. It will perhaps add a lot of energy to the program. Can we do these 2 products, if not others, together with you? Let that be the test case. You see, doing things is one thing, having a potential is another thing. So we have the potential to do things in 100 days, but can we demonstrate our capability for 1 or more of these products? So that's my specific request, but broad request in this particular point is that we will be expected to deliver products with speed and at a scale. And therefore, let that also be one of the defining principles over which we should build our relationship. I'm so happy to note that we have 120 more acres of land. I didn't know it. So you come and set up your R&D shops here. Let's design that facility in such a way. This could be the game changer for future. For some of the things that I'm talking about, this could make all the difference. Imagine where this institution is leading the nation with your support. So please come and make a THSTI. आप यहाँ आइए, हमारे साथ बनाइए, जुड़ जाओ, लॉन्ग टर्म रिलेशनशिप में जाओ. जो यहाँ आज नहीं है, उनको भी बताइए कि कुछ अलग हो रहा है यहाँ पे अब. ठीक है, वो सिर्फ हैदराबाद में या और उधर नहीं हो रहा, अब यहाँ भी हो रहा है. और आप प्लीज जुड़िएगा. एंड फुल सपोर्ट है, सेक्रेटरी साहब हैं यहाँ पे. वी आर ऑल देयर एज ग्लू फ्रॉम नीति. वट एवर बेस्ट आई कैन डू, we will be very, very happy to do. This is the policy of the government and we are committed to do it and we must make it happen. My last point is to the students here. Can't help being a teacher. A teacher is a teacher all the way. So I just want to tell you, a simple advice that I have been giving to my students, and that's about excellence. This institution, this hall, this cluster represents India's biotechnology, science excellence in a way, and you are going to raise the level of that excellence further. What I wish to tell you is that excellence is different from the business as usual, and excellence is different from what you can do, but it is what you actually do on a daily basis. Here is my favorite quote. It is attributed to Aristotle, but this is what it is. I quote, "We are what we do repeatedly." This is for students. All of you already know that. We are what we do repeatedly. Excellence, therefore, is not an act, but a habit. Unquote. आपको ये बताना चाह रहा हूं कि पोटेंशियल होता है कि मैं क्या कर सकता हूं, कितनी दौड़ तेज लगा सकता हूं, कितना अच्छा लिख सकता हूं, कितनी अच्छी तरह से समझा सकता हूं, एक्सपेरिमेंट कर सकता हूं. पर फर्क यह है कि क्या वो ऐसे ही रोज काम आवश्यक करते हैं. आदत है उस तरह से बेस्ट काम जो पोटेंशियल है आप काम करने की कि नहीं है. So I want us to make excellence as a habit. And my clinical, experience, or shall we say, system. So we would have residents who would write very nice prescription and write notes and case sheets when they know that a consultant or the head of the department is going to take a round. तो बड़े ही उस दिन अच्छा दिखाई देता है. लेकिन रूटीन में तो ऐसा लिखते हैं प्रिस्क्रिप्शन, वो पढ़ी नहीं जाती, ना मुझे से पढ़ी जाती है, ना पेशेंट से, ना फार्मासिस्ट से. कहने का मतलब ये है कि पोटेंशियल तो आपका था, लेकिन आपने काम तो रोज ऐसे नहीं किया. आप एक्सपेरिमेंट करते हैं, कहीं शॉर्टकट्स लेते होंगे कभी कभी, हो सकता है नहीं लेते होंगे. हम तो THSTI में हैं. कहने का मतलब यह है कि अगर आप एक्सीलेंस को हैबिट बनाएंगे तब ये देश ऊपर होगा और आप आगे बढ़ेंगे. कभी कभी उसको अच्छा करना जो है वो आपका पोटेंशियल है. पोटेंशियल इज डिफरेंट फ्रॉम अचीवमेंट. And therefore, I urge you that in your daily life, students, scholars, please adhere to excellence. What you write, WhatsApp पे भी जो मैसेज लिखो तो उसमें कॉमा और कैपिटल लिखने की आदत होगी, तभी जिस दिन आपका PhD का वाइवा होगा लिखा जाएगा तभी ठीक होगा. इसको आदत बनाइए. रेफरेंस को मैं बाद में तरीके से लिखूंगा, उससे नहीं होता, वो कभी अच्छा नहीं बनेगा, तब टाइम का प्रेशर होता. सो मेक एक्सीलेंस एज अ हैबिट and ensure that the high values, excellence has to go with values of integrity, values of purity and truth, because science is a pursuit of truth. Thank you very much. Jai Hind.
Thank you, sir. I now request Professor Karthikeyan to kindly felicitate Professor Vinod Kumar Paul. Thank you, sir. We begin with our next session, Industry Speaks. I now hand over to Dr. Gaurav Batra and Dr. Manish Diwan to kindly conduct the interactive session.
Thank you for the patient listening, all industry folks and friends. We've been hearing what the Institute is doing, and we also heard about the commitment from Government of India, Department of Biotechnology, NITI Aayog. Thank you so much for that. So I would open the floor and request our industry people who have gathered here to reflect your thoughts briefly. If you want to say something, have some bit of advice for us, or you think there is some potential that we can take it together. So over to you. Anybody who wants to go first may raise a hand. Dr. Gaurav is here. And
So any suggestion is welcome.
Hi, everyone. Good afternoon. This has been a very inspiring session. Thank you to the speakers.
May I request if you can introduce yourself first? Thank you.
I'm Abhinav. I'm from a company called Sphera Pharma. We are into small molecule-based drug discovery and development. And it's been a very inspiring afternoon session. Thank you to everyone who took the stage and shared their thoughts with us. Being in this space now for about 15 plus years, operating in India, we realize that it is an uphill task. Any form of innovation is an uphill task. And it's very well said that the environment has never been better to be able to do something. And in that zone, as we work with academic partners such as yourself, such as THSTI and other DBT institutions, as industry, we are not afraid of using the words profit. And we don't want that to be a taboo word. The question is, how can we embrace responsible profits, keeping innovation and affordable patient outcomes at the forefront? And with institutes like THSTI, my question is, how can we create a framework where we can partner with academic innovators, professors, and PIs, and be able to create corporations and biotechs that can be the Gilead or Genentechs of the future? Thanks.
All right. We will try to assimilate some of these questions. And maybe I think once we get into individual PI's laboratory, some of those questions can get addressed. But I think just to pick up what you said, there are multiple ways of engaging. Either, as we earlier said, it could be program centric, one particular assignment. There could be a larger long-term commitment. There could be joint projects. There could be physical footprint here, picking up or sponsoring students, PhD scholars, creating innovation lab here, creating faculty positions here. So there can be n number. Maybe if you have anything in mind, you can tell us. And if you have some leads, for example, or hits, you want to get it characterized using the facilities here and expertise. I think those co-developments can happen. Service models can be seen. So I think as we heard about the capacities and facilities plus expertise, I think it's open, and the director is fairly open. The administration is also very conducive. There is a go-ahead from DBT. So I think it's an open playing ground. So help define the rules. And I think during the process, we will iterate and also learn some of these practices, then we can adopt it for a longer term.
Hello, Dr. Dhawan. So 200 acres land, all
So
right?
maybe rule of the game, kindly tell your name
So
and the
I'm Dr.
company.
Rajkumar Haldar. So I'm CEO and founder of Juvenile Biomedical. So it is a very interesting project. But whenever you're comparing with Boston, all those kind of things, no? And if you want to have a lot of industry over here, you need to have much bigger space. Just keeping the building and productivity will not happen. You need also a lot of entertainment, all those kind of things. Because most of the cases, innovation happen when there is entertainment. Otherwise, it will not happen. Cultural event. 200 acres land and asking for industry to invest, I think that is very less. Second question is, will you give the space or you will give the building? Okay, and this is how long it is. Let's say we want to invest 10 crores, 20 crores, 30 crores within the next 2-3 years. Will you give that space for next 30 years? Because to build the industry, or to build a product and to be sustained on that, it's huge amount of time and patience. Third question is, whenever you see that you have the stamina, or you have a lot of people, 800 plus students you are saying, am I right? How many students are staying for a long time? How many faculties are staying for a long time? Even you are doing some clinical trial or anything. After maybe 2 years, most of the people are leaving. Then what about the data? So these are the concerns I do have it, but I am highly interested to invest in THSTI. And I also want to be part of this biopath if you give the right opportunity. So we are not just nothing but a startup company. We started with the BIRAC big project and we want to give back to the society. So I think this time you also have to, you have to hold our hand. And for long term, that is, I think Dr. Rajesh Gokhale, he also said it, make it happen and make the country proud. Thank you.
So a lot of questions built in. I think since you heard all of us in the morning session, maybe you can put up a expectation proposal to us. Tell us what is in your mind. Is 10 years long enough, or you need 33 years lease area? So you tell us what you have in mind. I think then we can take a cognizant decision and discuss internally, see what evolves out of what we see together. Yeah, thank you. Yeah, you have a mic right next to us.
Dr. Diwan, thank you very much once more for the kind invitation. My name is Dr. Anand from Singapore, Govin Capital.
अभी सबको याद हो गया.
अभी आपने रूल बनाया तो मैंने सोचा I follow the rule.
Thank you so much.
Thank you.
Yeah.
What I do in Singapore is gap funding. So what is the meaning of gap funding? As you know, I spent 4 years of my career with A-Star and 10 years of my career with the government of Singapore, promoting life sciences ecosystem there. At A-Star, we have an industry gap funding. What it means is that project by project, the kind of work that you have shown today is high quality work. Maybe some of the projects, if you can invite the industry to come forward to fund at an early stage to make them industry ready. Because as the scientists were speaking today, what I saw is that some of them are industry ready and some of them will become industry ready. So those projects which can become industry ready, if you can invite investors or industry to co-invest with this, with the clause that they get a right of first refusal to take it forward towards commercialization, this can be a viable model to work with institutes like THSTI and others. That's number one. Number two is, I repeat, we need to formulate a policy for academic spin-offs because any government cannot fund forever a research project. There has to be what is called as a return on investment. And the return on investment is possible when you invite third parties to come and invest capital in spin-offs. So we need to formulate policies to facilitate scientists for academic spin-offs that can create economic value and bring in third-party money. So it is a win-win for both the institute as well as for the third-party investor. Thank you.
I think both are valuable suggestions. In the after lunch, when we have got some glucose in, we will go to individual PIs. And I think that is the objective of that interaction. They can then tell that these are the leads in their mind which are ready based on merit and perspective of industry. You get to see those. And I think there the propositions of co-development or polishing it up further, enhancing it through the TRL journey, that can evolve. And I think once industry people speak one-on-one with them, I think we will mature those initial collaboration leads. To the second point that you mentioned, I think Dr. Karthikeyan and Dr. Gokhale are keen that these things can get facilitated. And I think everybody is hearing. So policy level workout we can do at the back end. And NCR cluster would eventually inherently should have all these things in place. So we will work on that so that there is a clarified path for faculty people to kind of move that side.
I am happy to commit 5 crores to THSTI for the projects for commercialization, with the caveat that Government of India, the decision makers who are here, will support us with at least 2 rupees for every 1 rupee that I put in. Thank you very much. That's my humble request.
Sure, sure.
Yeah.
I think we will put that on record. Thank you, Anand.
Yeah.
Thank you.
Thanks for this fantastic session. This is Syed from Tech Invention. We work in the space of infectious diseases. My question is, since most of the emerging diseases, infectious diseases are zoonotic, and most of the platforms are also applicable to ruminants, livestock, where the need is there, would the THSTI continue to maintain its focus on humans or also look at animal medical countermeasures?
I think we have expertise that we initiated with, which is more human centric. I think the life sciences thing cut across, and One Health is one of the national mandate. So if I may say that we will also extend our expertise and facilities for the benefit of the
industry.
Yeah, of course.
And the focus is on One Health somewhere. So all those what you are asking will come under that. So yes, we are to largely to that side. Yes.
Yeah,
Perfect. Thanks.
no restrictions that way. Yeah. Absolutely.
Thank you, Manish. It has been inspiring to be here, although I came late.
Sir, you
My
are
name
from
is,
the
sorry,
pharma
my,
side.
my, SYNCHN. I'm the Secretary General of Indian Pharma Alliance, which represent 23 top companies, 60% of domestic sales, 85% of R&D expenditure in India. It is inspiring to be present here, and thank you, Manish. It was great to hear Dr. Vinod Paul, and just going back to the journey of vaccines, it gives us confidence that we can do it and we can make it happen. We are very good in generics today. 1/3 of the global market is generic, but 2/3 is the space where we are not there. And this initiative of upgrading the value chain is fundamental going forward. Dr. Vinod Paul talked about PRIP scheme. Industry academic collaboration is fundamental. Regulatory reforms is fundamental. But speed and scale will be very, very important. This is one of the top topics with all CEOs of the company. And Manish, we will come back to you. We will get all of them here, and we will take this agenda forward, because there is no choice for us but to move forward in this agenda.
Thank you very much, sir. I would add, I think those people who have taken time out and come here on this today's interaction, I think all of you become our ambassadors for good science or good quality, whatever you imbibe and you evaluate in your own perspective, you would be speaking to in your networks and your counterparts. So we look forward, and we will go and support that whatever you engage with. And if you require our interjections, our presence over there, or we can bring a second round of industry meet and bring them in person there. Thank you. I think Mahesh is over there.
Hi, Mahesh Palgat, CEO Group of Vida Clinical. I think this is fantastic what Sudarshan mentioned. I can add more to it, that this has actually been a very, very enlightening thing. The one thing that I think we need to recognize that is unique here in this opportunity, at least for us who are getting exposed to this for the first time, is there's a disparate group of technologies, capabilities, projects that are happening. One of the things that we haven't done historically as well in this ecosystem is bring data together. Today, we know the value of data. We know the value of data in many, many, many different ways. If we can start looking at this as how do we want to build data platforms that can help us rapid decision making. We know AI is driving a lot of discovery-based technologies. We know AI is getting into clinical research. We know we are looking at even things like virtual patients, so on and so forth, right? How is this opportunity of putting so much science in one place going to help us build data and therefore drive decisions through data? So I think that's something to think about. But wonderful opportunity. Thank you very much.
Thanks, Mahesh.
Yeah, so at THSTI, we have whole program on data. We have data management center. We have AI-driven. We are developing AI-driven tools and combining all the expertise which we have in-house. So we are working in that direction. But thanks for bringing this again.
So as we mentioned, there is a connectivity center, which is like Indian Biological Data Center. So at least we are being sensitized and echoing what you are saying. So that data is getting collated and placed there, which would be accessible to all of us as we speak about it. There is also INSA-GOG, that story. So that data also becomes accessible, and it comes within the BRIC network. So it's powerful, I think, cluster that way. Because not only you can leverage what we produce here as a scientists and scholars, but it is pan-India, all the BRIC institutions together. They can also, that knowledge can also be leveraged. Thanks.
Yeah, thank you. My name is Professor Samir Kulkarni from ICT, Institute of Chemical Technology in Mumbai, heading the Department of Biological Sciences and Biotech over there. Now, I mean, I've been part of the industry, in the biopharma industry, worked in Intas and Dr. Reddy's before for about 20 plus years. One of the things which I heard quite a bit today from different people from TESTI was about and I think, Dr. Manish, you also mentioned that basically there are plans to set up GMP facilities into this particular place. I think one of the things that perhaps can be leveraged or can be requested to the industry is also to bring in the know-how. Because like what, I think Dr. Samal said, right, that the scale of operation is for the lab and then eventually to take it into the industry, the processing and all that. And it's not just about the scaling up, it's also about how to scale up properly to bring in the GMP practices. Now, GMP practices is something which the academia is going to lack in its experience because they have never worked in those particular facilities. And that is something which perhaps the industry can contribute by maybe where BIRAC or DBT can actually create some sort of an expert group wherein this particular feature or this particular aspect can be brought in properly, methodically, because as all of us know, a lot of the pharma companies are facing issues, in terms of US FDA is coming in and telling us that there is nothing wrong with the product, but there is something wrong with the practices in which you are making the product. And this is something which I think, could be a very potential gap that might come up, if that particular thing is not really thought out. And I would just like to bring that, draw the attention to that specific feature here.
Absolutely, Samir. I think very valid point. We don't have that experience in general. So that development experience is of course lacking, and it can only happen. With industry coming closer to us. Nobody in the institute or through NCR cluster, we are not saying we are getting there to manufacture a product. We don't have that capacity, neither we want to build that capacity in the cluster. We want to build capacities and promote pre-product development. And to do that efficiently, we want to work closely with the industry. And I think your point is absolutely valid. Just to give one piece of information, the Medical Research Center would be coming up fairly soon. So come September, we will have the cold shell ready. By let's say January next year, we are seeing that the infrastructure would all be laid out. And that is where one of the clean room facilities and the GMP is also envisaged. So I invite all of you, whosoever thinks that cell and gene therapy is of interest and you can help us guide, advise, come together with us, and then we can build it together so that we don't miss out on anything which we think we know through the experts group that we engage. We will be very happy to engage with all of you. Yeah, thanks. I think there's one thought
Hi,
here.
this is Vikas, and I come in from Origin. We are a CDMO. And I guess just to take forward what was said before, we definitely, as an industry, can offer the expertise on GMP. So please feel free to get us involved there. We definitely can bring in the expertise that we have.
So I think I would like to make a point here that often it happens that we said we are happy to engage. You said I am happy to if I've reached out. So वो बात खत्म हो जाता है. So no, like both sides, right? So I would suggest that in case you consider that this could be an extension center. You have the local infrastructure in your company, but you would not be using it for every single thing, right? Some exploratory studies or something that you can think of can be kind of offshore. So consider this place as that window. And there, I think the interest of the two parties could match, and then we can prolong that and continue an engagement.
No,
over there.
definitely, because I guess it has to be a two-way street.
Yes.
Some of the work we do at our end, some of the work we get done out here.
Correct.
And I guess that interaction over a period of time would yield the synergies that we are looking at.
Absolutely. So train us. All I'm saying is
definitely would train you and get
Yeah.
trained too on
Yeah.
certain things that you guys are an expert on.
Great.
So I think that's something that we need to set up wherein the connect is not limited to gathering like this, but gets happened over a period of time because of
Right.
collaboration. The intensity could go up as we work together, but at least let's make a small beginning in terms of the interaction. And I'm sure it's going to ramp up over a period of time.
Certainly. Thank you. Thank you. Dear.
Hi.
There is some voice, yeah.
Yeah. Hi, this is Vaishali. I'm a senior GM in SYNCHN. So I thought this is a good time to talk about the China Plus One strategy. So as we are thinking of building GMP facility, and I was really elated to see that the GMP facility is getting built, we talked about the golden triangle between the industry, the academia, and government. I think just to comment that this is the right time to hold hands because the world is looking at India as our, alternate supply chain. Considering that, this is the time to act. So I wanted to make that comment.
Thank you. I think I appreciate that. Yeah, we echo that. If you can pass the mic on the far end, that's all.
Good afternoon, all. And first of all, I'd like to thanks THSTI in terms of organizing such a wonderful event. I'm Saurabh Sachdeva, I'm VP for Polyamina Nanotech. We are on a mission to reduce the acquired infections over the globe through hand to multi-pathogens. So I'm not a subject matter expert as the things which were going on, so I was trying to learn and gauge the things. But two questions in my mind. One is, over the last 12 months, as an head of THSTI, what one thing you think you have to unlearn when you move towards to a 16th Foundation Day will be happening next year? And as we make New Year resolutions on every year, what resolution you will be making on tomorrow for THSTI?
Manish has put me on the spot here. So, like he said earlier, I think, and like many of you said, this is the time to start whatever we are doing. And this is an inflection point that we will take advantage of. Now, all of you, you want something also from from academia. So if you express those interests more in not just saying here, but in more concrete terms, then we can certainly act upon them, and we'll see how it works. I mean, there will be also some give and take, and we will decide what is the way forward. And we are all very interested in this, and the next year, SYNCHN will be a more international one. We'll probably get more international, not only more national, but more international as well. This is going to be a much bigger event next year. And we will hopefully have a much bigger faculty as well at that point in time, and we'll be doing much more work. So I can promise you that.
Thank you, Dr. Karthik. I think whatever seeds we sow today, and I think that is with guidance of all of you. Tomorrow, I think it would grow even bigger. So we can unlearn some of the things. But I think as we interact closely with you, let us know that these are the better ways of doing things. Maybe this direction is more promising, more challenging. Let's go together and we can walk farther in the road there. Yeah. Ajay.
Hi. Hi, I am Ajay from Pursued Life Sciences. We are basically a startup. And being in this industry for last 5 years as a startup, I think. So what I'm missing here in North region. So I'm happy to hear that you are coming up with medical research center. But I think it's very important to have chemical research center also here in this cluster, because we do have there in Hyderabad and other cities. But having here in North, that is very much required, because any basic research we are doing, we do need chemistry. That is very, very important. That is one request.
I think that's a good point.
Yeah. And the second point, what I want to say that one, AI is coming very, very good way. And if I share about our collaboration with the other academic partner, we are doing partnership with, outside universities only for AI. And I don't see any good AI hub for pharmaceutical research in India. So that is other point we need to have.
Okay,
Yeah.
got it. I think we are conscious about AI integration. I think mother and child care data is sitting with 12,000 subjects, and we want to develop that gestational model in practice. I think Kshitij is also here. Philips is one place we have reached out for ultrasound images that can be gauged. I think, but there is there is plenty of opportunity for both discovery as well as from data science. And as Dr. Alka mentioned, we do intend to make biomanufacturing AI hubs, which is all data coming together and uh, uh, probing that and triaging and extracting what models and benefits we can do, and short scale the longish timeline for discovery.
And other point here that, during the COVID time, we did learn something like we were doing very good in the vaccine research, but we could not do anything in small chemical. I am literally saying anything means we didn't say anything coming up, from any academic or industry left small molecule. That main problem was that we don't have any chemical compound library, archive here in India. And I do see several institute working on different medicinal chemistry labs across the academic, or you would say CSIR labs there. But I don't see a comprehensive, some archive if you want to screen compounds against any disease that could be pandemic or what. So having that kind of archive will really helpful.
We'll keep that in mind. Let's see. Let's deliberate.
Yeah,
little bit
and
on
you
that.
can, during the breakout session, you can discuss about this particular issue with the team.
Yeah. So maybe we can pick up two more reflections before we break for lunch.
Yes. So my name is Khalid. I am Chief Scientific Officer at Panacea Biotech. And congratulations to whoever thought or conceived this event. I think this has been amazing. We're already collaborating with THSTI on no less than 5 projects. So we are very much there. And we will be very happy to further expand
What's your
our
feedback
collaboration.
like? You can tell the peers.
Feedback has been great. So we have collaborated with several institutes, and our interaction with THSTI has been so far one of the best, if not the best. So we have collaborated with several
If you
academic
turn
institutes.
around, these are the people this is the team who has contributed.
Exactly.
So some
Yeah.
of them would graduate, I think, in
Yes.
some time.
Yes.
And maybe they are looking for industry placement too, but not today. Not today.
Okay. So
do come back here.
So I was saying we'll be very happy to expanding our collaboration further. And the points which you mentioned about having facilities, especially R&D facilities here, it's very, very interesting. But I think the commitment has to be a really long-term commitment. 20 years, because if we start building a facility today, by the time we get up and running, it will be 3 years. And if there is a policy change after our facility is built, then we won't be in a good place. But this is really interesting. And I think if in further communication from your side, if you can elaborate on modalities how that can be, implemented, that will be great. You mentioned cold shell, warm shell, greenfield, brownfield. It could be anything, but I think that will be great. So thank you for this.
We are working on those finer details.
Yeah.
And sooner we get that through, we will actually publish it and put that up so that and inviting specific proposals under specific domain areas and open it up. So maybe bear with us for some time. But in the interim, we are happy to hear back from you. If you have a suggestion, for example, 10 years is too short, 20 years is too short, I want 33 years lease, a kind of commitment so that I can invest this much to create a greenfield project. So do let us know. And I think we are
Exactly.
Yeah.
That would depend on the modalities. And we are one of the few major biotechs based in Northern India. So we are very happy. I think so far we were jealous of Hyderabad, but I think soon we will be able to compete. Thank you.
Yeah, Dr. Manish, if I can come now.
Go ahead.
Yeah.
Maybe Shiva, you are the last guy, right? After him.
I'm sorry, I really don't want to stand between lunch, for everybody. Most of the people must be very, very hungry. So Dr. Diwan, I'm Agnibi from SYNCHN International, and I have a very quick and very short question. This is about any regulatory facilitation idea, which, this particular cluster is also thinking about. And this kind of a cluster will be also dabbling with a lot of latest science and technology, whether it's CAR T cells or bispecifics or anything. So the regulators have to also move really forward and really have to understand science and really have to do those clearance processes in time. And any kind of facilitation of those sort will be really helpful, I think, for this.
I think that's a very relevant pain point. And what we have envisaged is through BIREC, we have created regulatory information facilitation cell in Venture Center Pune. That's one. Since we are here, closed door to CDSCO, we are envisaging that we can have such offices, nodal offices, or in regional centers here in the cluster. Because if you're talking about 200-acre campus, large number of startups, SMEs, and industry players working on projects which are actually moving towards product development with industry. So a regulatory facilitation cell or a closer interaction with them in-house in the center, that is envisaged. So we'll try and impress upon CDSCO if they want to do it, BIS, FSSAI, and related regulators. DBT is anyways next door. So I think that element, we want to bring it here, along with IP, legal services, investors having their local offices here. So I think, and as you pointed out, it is relevant, yes.
Absolutely. In this particular area, even if you are also going ahead for, animal experiments as well, somewhere we can do something on primates, because I mean, those kind of facilities are really lacking in. So
there is a primate center coming up.
Okay.
There's India's only ferret facility, which is next door. I think I would encourage, what we have done is, in the interest of the time, we have moved the cluster tour later, after the lunch, after you have your food. Then I think before going for one-on-one PI interaction, you can assemble here at 2:30 after lunch. So we will take you to that particular PI group, but through a shorter cluster tour, and then we take you all the way to the PI. So you can have a glimpse of all those facilities outside. Anything you think you are interested in, you can do a deep dive, and the facility in charge or PI would be happy to interact with you. Okay. Shiva.
Hello, everybody. My name is Shiva Natarajan. I'm the interim managing director for Miltony Biotech in India. So Dr. Manish, Dr. Karthikeyan, I can't thank you enough for this opportunity. It gave us a brilliant window of what you're doing over here. We walk away inspired and enriched from the work that you're doing. I'll also take a bit as a lesson from what I go take as a message from here. One slide from Dr. Karthikeyan, which said that India is just 3 billion when it comes to healthcare investment, whereas globally it is 245 billion by another country. That is something that we need to change. I take that as a message. The second will be from Dr. Amit Awasthi's session, where we specialize in CAR T and we are amplifying our CAR T presence in India. There are 6 CAR Ts in the world and 5 of them use our platform. So how fast we can collaborate is something that I want to take home from here. I like the part that we mentioned about the velocity of what we need to do in India. I think in the last 1 month that we met, we are already closer to getting into a very good collaboration with THSTI, and we are very proud of that. What we can offer on board will be the skill set that you mentioned. So that is something as a German company, we can bring in skill into the company, into the country, to THSTI in particular. And the next one on scalability, how fast we can scale up, that is something that we'll be willing to offer, right? So thank you so much for that.
Wonderful. Thank you, Shiva, for reiterating that. I think the administration and the management support is clear. So I think tomorrow we'll be signing or getting into agreement with some of you who are present here. So we look forward to taking that a long way through. Thank you.
Hello, hi, sir. Maybe the last question, sir. Priya from Tizal Biopark. Here.
Okay.
Hi, sir.
Yes.
Hi, I'm representing Tizal Biopark. Along with me, managing director and the managers that came here. So Tizal Biopark, we have around 64 industries in Tamil Nadu in Tizal Biopark. And we also have a Tier 2 city in Coimbatore we have established. So the one thing is really TI is doing a very great job. So if there is any kind of validation center, maybe from and also to access the mentoring pool from THSTI, it will be very much useful for our startups.
I think this place is a convergence center, as I repeatedly said.
Yeah.
So it is not limited to only outcome from the THSTI. So the NCR cluster is a national asset, common access facilities, all the TRC, MRC that we spoke about, which are now coming into being within next year. So those can be leveraged by academic leads or startups and SMEs and working closely with the NCR cluster. I think feel free to identify which is the right expertise which is locally here and get engaged and let your startups and SMEs come forward.
Sure. Thank you.
Thank you.
I thank all the participants for an engaging discussion and all the suggestions provided by our industry representatives. We'll have a small photo session. I request all of you to be kindly seated, but
Yeah, the photographer will be on the stage and you guys
taking
will be
the
taking photograph from
photographs.
that only.
Acha. As Dr. Manish Devan already set out the instructions for the breakout session and the guided campus tour. So we'll all assemble around 2:30. I request all the industry representatives to assemble here after the lunch for the guided tour, and our set volunteers will guide you. But before that, I request Dr. Amit Pandey to kindly convey our vote of thanks to you all.
So a very good afternoon to you all. On behalf of BRIC THSTI family, I would like to express our deepest gratitude to all of you for making this industry meet a resounding success. We are honored to have had such distinguished guests grace this event with their presence. Your invaluable insight and expertise have been immensely inspiring to all of us. A very special thanks to Professor Paul, the chief guest. I want to share an anecdote with him. I was honored to, escort him in one of the previous events. And I was very naive. I said, sir, we are missing Professor Bhan today. So his immediate response was, you are missing him today. I miss him every hour, every day. So thank you for making yourself available and guiding us. So Professor Ganguly, we all know that you have been instrumental in shaping this cluster. We look forward to your guidance in future, and thanks for coming. Professor Gokhale, everybody knows his scientific and entrepreneurial story inspires all of us. And, we are here because of the stories that we have heard of his success in science and entrepreneurships. Thank you. Thank you for coming today, and we are looking forward to your leadership. I thank Dr. Alka Sharma. She has been fighting for THSTI. Thank you, madam, for speaking on our behalf in every forum. We really value your, help. So to our industrial partners, we are also grateful for them for their active participation and collaborations. We look forward to a productive dialogue leading to fruitful research collaboration between BRIC THSTI and the industry. My sincere thanks to faculty, scientists, administrative staff, students, and volunteers of THSTI for tireless effort in organizing this event. For conception to education have been truly commendable. Finally, a big thank you to all the attendees for your active participation and stimulated discussion. Your presence and enthusiasm have made SYNCHN 2024 a truly enriching experience for everyone involved. We believe that the collaboration and partnership forged during this meet will pave the way for future advance in human health. Finally, I would like to thank our Executive Director for helping us organize this event, and we really look forward to his leadership. So finally, thank you once again for accepting our invitation and contributing overwhelming to the success of this event on a Sunday. So thank you. A big thank you to all.